no nace como una universidad docente únicamente, es el corazón de las universidades, la docencia. Pero desde sus orígenes nace con una inquietud de lo que hoy se conoce como universidad compleja. Proyecto de universidad compleja que el rector fundador Morales lo sustenta, pero que luego el rector Félix Martínez Bonatti le da esta complejidad a la universidad, es decir, no es solo formar profesionales, como decía él, de Chile al mundo, o de Valdivia al mundo, sino que también formar científicos, hacer ciencia, y las artes y la ciencia ha estado en esta universidad, de la Universidad Austral de Chile, desde sus orígenes como parte fundante e una inquietud esencial. Por tanto, quiero comenzar destacando muy especialmente el trabajo que ha impulsado el Instituto Milenio en inmunología e inmunoterapia, que se ha consolidado como un aporte muy significativo para la investigación y la generación de conocimiento en nuestro país, y han demostrado que es posible desde Chile desarrollar y patentar soluciones inmunológicas propias. Esto es muy valioso y nos posiciona como un país donde podemos hacer ciencia de excelencia, a pesar de todas las restricciones que podamos y que siempre vamos a encontrar en todo el ámbito del conocimiento. Yo vengo de las ciencias sociales, de la administración pública, ciencia política, y si uno quiere hacer una competencia de encontrar restricciones, pues en cualquier área del conocimiento vamos a estar ahí peleando quién tiene más restricciones, pero se avanza. Por esto mismo es que quiero manifestarles mi alegría y nuestra alegría como equipo de rectoría de que este encuentro tenga lugar acá en Valdivia. Y qué bueno que el doctor Calvo lo invitó a la CUN. Me alegra mucho eso, que se haga también ese tipo de... La CUN tiene una historia bien particular, doctor Calergi, ¿no? Puede ser motivo de investigación de otra área del conocimiento, pero tiene un origen bien particular también en procesos de innovación. La cerveza que hoy día toma, y hoy día hay más de 20, 30 variedades de cerveza, no es solo la CUN. Así que ojalá que hoy día doctor Calvo lo lleve a otro lado, que pruebe otra variedad de cerveza. Las experiencias recientes con la pandemia COVID-19 han demostrado la importancia de contar con comunidades científicas interdisciplinares como la que es expresión este Instituto Milenio, conectadas y preparadas para enfrentar, en el caso de la pandemia, efectos pandémicos y actuar articuladamente para aportar a las decisiones de salud pública con datos y evidencia relevante en los momentos más críticos. La inmunología ha demostrado ser un campo fundamental para comprender y enfrentar amenazas sanitarias presentes y futuras, y al mismo tiempo nos recuerda que la investigación tiene un impacto real en la vida de las personas cuando logra así traducirse en nuevas formas de comprender, prevenir y tratar enfermedades. En este sentido, este simposio refleja ese valor de la colaboración entre universidades, centros de investigación y el ámbito clínico para acelerar el desarrollo de nuevas soluciones biomédicas. Para esto es clave el desarrollo de un ecosistema científico nacional integrado, insisto, como el que refleja este instituto, que ponga en valor espacios como este que inspiren y desafíen a las nuevas generaciones de científicos y científicas. Encuentros como este no solo enriquecen el diálogo académico, sino también tienen el potencial de transformar vocaciones y proyectarlas hacia el futuro. Les doy entonces la más cordial bienvenida a este simposio. Disfruten Valdivia, disfruten la universidad, hay un rico café ahí en el Jardín Botánico, no sé si ya lo han visitado. Café, no sé de dónde es, uno es colombiano y el otro viene de Sudáfrica el café, pero es muy bueno, cualquiera de los dos. Disfruten el Jardín Botánico, la belleza de Valdivia, la belleza de la universidad. Estamos comenzando las clases, estamos comenzando con mucho ánimo, y agradezco de verdad sinceramente su presencia y les deseo una jornada muy provechosa de reflexión, intercambio y proyección para la ciencia que se desarrolla en Chile. Como le dije a Mario y a Alexis, yo estoy en el gabinete, termino de intervenir y me voy a ir de inmediato a continuar con las labores mundanas de la gestión universitaria, entre otras cosas, desarrollo de la ciencia, pero también de la gestión y el bienestar de los trabajadores. Así que me voy para allá. Muchas gracias por esta invitación. Quise venir acá a saludarlos y desearles que tengan un exitoso simposio. Muchas gracias.
no pasa casi nunca, vamos adelantados. Así que voy a consultar exactamente por el asunto de la transmisión a qué hora comenzamos. ¿Comenzamos de allá o esperamos a las 9:30? Ya. Empezamos de acá entonces. Ya. Nosotros vamos a tener que cambiar el idioma ahora para los asuntos de la transmisión. Then I will present to Dr. Alexis Galerkis. He's full professor of Facultad de Ciencias Biológicas, Pontificia Universidad Católica de Chile, Director of the Millennium Institute on Immunology and Immunotherapy, Santiago, Chile. He will present the talk, Scientific Basis for Vaccine-Induced Immunity that Prevents Acute Illness and Cognitive Impairment Caused by Respiratory Viruses.
Todo el tiempo que he ganado estoy tratando de perder ahora. ¿Se me va a mandar el link para conectarme a Teams o es Zoom? ¿El que usted inició, entonces? hacer esto acá en el Teams, ¿no? ¿Se ve? Ok, muchas gracias por la paciencia de la introducción. Y bueno, agradecer a la organización de este simposio internacional en inmunología de la información. So, okay. Thank you very much, Dr. Bueno. I will switch to English because I think that's the official language of this symposium. So now that we see everything in the so now I'm seeing here the wrong thing. Muestran el Teams, lo opuesto lo que muestra acá. Entonces, no sé muy bien cómo se puede. Ahora. Ahora. Sí. Tenemos que funcionar 3. Bien, entonces, ¿vamos? Okay, so I'll switch to English now. I'll talk today about the work that we've been doing on vaccines and the way they induce immunity and how that immunity can prevent disease. caused by infections as well as other sequelae, like cognitive sequelae. So, but viruses have been around for many, many years. So this is an example. Smallpox is a disease that's caused by a virus, a DNA virus that's not there anymore. Susan, is this work? Can I switch to this one? Hello.
Sí.
Sí. ¿Este me lo hago? No, prefiero jugar este. Entonces, ¿se escucha con este micrófono? Sí, perfecto. Gracias. Entonces, so the disease caused by viruses has been around for many years. So smallpox is an example, and thanks to the development of vaccines, this disease was eradicated by the late 1970s. And this is the only one example that medicine and science working together have provided to control and eradicate the disease. Other diseases like polio, measles, rubella, and mumps have been also controlled thanks to vaccines. As you see, every time we have the incorporation of vaccine strategy on the population, there is a significant reduction in the number of cases of disease. And as long as vaccines are being used, this is the case. The problem we've been seeing in the past years, recent years, for measles, for example, is the reduction in coverage of vaccine usage led to the increase of cases in countries like the US, some European countries, and also in Latin American countries like Argentina. So it's important to keep passing the message how vaccines can be safe and efficacious at preventing the disease. So we can see that when vaccine coverage goes up, then these are several different vaccines. The number of cases for all those diseases that are prevented by these vaccines go down. So it's a very clear effect, very clear positive effect of vaccines. And we have a major challenge in countries like Chile because there is a gap between industrialized countries and developed countries in terms of the access, the quality, the availability of vaccines for human use. So that poses a very significant challenge for us, and it's important to connect from academia with the industry, because we're collaborating with Pfizer in terms of promoting the access of vaccines to people in countries like in Chile. And in our region, in Latin America, only 4 countries are able to produce vaccines. Those are Argentina, Brazil, Cuba, and Mexico. But their capacity for producing vaccines is very limited. They can only cover for their own population, and sometimes can't even do that. So we've been working for the past 4 or 5 years on this project to try to gain some access to vaccine production here in Chile. And we got approval by the Chilean regulatory agency, the Instituto de Salud Pública, to build this lab that is meant to produce biological molecules and vaccines under GMP, or good manufacturer production conditions, according to the Chilean rules and regulations, and also we try to comply with the EMA and FDA. And together with the building of the lab and the purchase of equipment, we've been working on training people in collaboration with a center in Italy and another one in Greece, where they already have a GMP facility, because it's very important, the training of people that will run the lab. And that's been a lot of work, just to try to put that, just have that training complete. And these are some views of the lab. So this is the kind of a not so new, not so recent video, but just how the lab looks like. It's about 300 square meters, and we have all the equipment in place. And at the moment, we're finalizing the work with the HVAC system, which is the air conditioning system, which is very delicate, very complex. And that's the last step we are working on at the moment. And we already have submitted this to the regulatory agency for approval in Chile. Now going to the science that my lab does. So we study T cells, how they recognize antigens on dendritic cells, and this type of response is required to have bacterial immunity as well as viral immunity. But we know that we get sick, most likely because these pathogens like bacteria and viruses are able to suppress and block the synapse and prevent T cell activation. And we found, and I'll show you that respiratory syncytial virus as well as other viruses have the ability to do that. So we study in my lab 4 different viruses, RSV, metapneumovirus, we have done some work with Hanta, and also recently, of course, with SARS-CoV-2. And for RSV, this is a major problem, still is, even if we have now a vaccine that was developed by Pfizer for pregnant women and an antibody that is being used in Chile. Even if that's the case, those technologies have contributed to reducing the viral burden in the population. Still, I think it's important to have a vaccine for the infant that can be directly used in them, not just by passive immunity. So we found, and we published this some years ago, that immune cells infected with RSV are not able to respond to antigen. So these are antigen activation assays, IL-2 production. You can see IL-2 production when the antigen is there, but if the cells are infected with RSV, there is no IL-2 production. And then we looked at the mechanism of inhibition and found that uninfected cells, they establish these contacts, synapses, as you see here, dendritic cells in red, T cells in green. When the cells are infected with RSV, there is a significant reduction in the number of synapses. And we calculated this to be about 67%. So we proposed a hypothesis that a protein from RSV, from the virus, goes to the membrane of the dendritic cell and prevents the establishment of this synapse. And went and checked each of the proteins that are encoded in the genome of this virus, which is 10 genes that encode for 11 proteins. So we went and tested each of them using different models. I'll show you one model. This model consists of planar lipid bilayers with lipids that have needles, so we can have his-type proteins to sort of reconstitute the antigen presentation, antigen presenting cell surface. So have antigen, addition molecules, and the different RSV proteins. And we come with live T cells that can react and respond to this stimulation. And we found that in absence of protein, we see clustering of ICAM and TCR. This is the control condition. When we have M2, which is an RSV protein, we still see clustering of ICAM and TCR. But when we use N, or the nucleocapsid of the virus, we see a significant reduction of the clustering of ICAM and even a change in the shape of the T cell. So then we did another assay. This is live cells. Here you see the TCR in red, the MAC or the antigen in green. And every time you see a yellow, like here, these are synapses. This is the same assay, but with the M protein. And that clearly shows there is a significant decrease in the frequency of this yellow overlay, which is a suggestion that the N protein is interfering with the synapse. So we also found that the N protein, as the F and G, which are the surface proteins of the viruses, the N protein is also located on the surface of infected cells. This is microscopy, and we did the same by flow cytometry, and found that the nucleoprotein is clearly playing a role by being the surface of infected cells. So when you find this kind of, if you make this kind of finding, then you can use this approach to create vaccines using that antigen as a target. So we went ahead and created vaccines against RSV, metapneumovirus, hantavirus, and SARS-CoV-2 using the nucleoprotein as the target antigen. And we use for that BCG, which is the vaccine used for tuberculosis. The reasons for using BCG is because the only vaccine has been approved over receptory for use in newborn babies, so it can provide immunity since birth. And BCG can prime cellular immunity, lead to some antibodies, and this is very important, it also leads to trained immunity, which is important for viral immunity. Susan, is the presentation going okay? Okay, good. Closer. All right. Okay, so we have characterized the way this vaccine works in terms of the immune response. We know it is able to prime T cells that produce gamma interferon that help CD8 cytotoxic cells and B cells to respond to the virus. And we the mechanism, PNAS and different other papers have a patent for the vaccine, and also produce a GMP lot or batch of this vaccine according to the FDA. This was made in the United States, and of course also complies with the Chilean authority. And the reason for using BCG is because it's been safely and massively used in humans for over a century. We can modify genetically to express antigens. It has a strong adjuvant capacity and is able to induce Th1 immunity, what is needed for an antiviral response. So we have tested this vaccine in 6-7 animal models. I'll show you only one of them, only a couple of them. So these are mice. Mice, when they are infected with RSV, they lose weight. And you see viral replication on the airways here in immunofluorescence. But when the mice are vaccinated, there's no lost weight, and the vaccine also prevents viral replication on the airways. These are MRIs for these animals, so these are the control, 2 mice, 2 infected mice. These are the foci of inflammation on the airways, and these are the vaccinated and infected. You see no inflammation on the airways. And in terms of the immune response, it triggers T cells that produce gamma interferon only in the presence of the RSV antigen, and also lead to the production of immunoglobulin that are specific for RSV, specifically IgG2A, which is beneficial and is higher than IgG1, which is thought to be detrimental. So when we presented this data to the FDA in the US for potential clinical trial, they suggested they were okay with doing assays on toddlers, meaning children that are seropositive, and adults. But in order to do trials in newborns, which is our final target population, they requested to evaluate first the vaccine on a newborn animal model. And we were lucky because RSV has 2 versions, human RSV and bovine RSV. So we collaborated with a colleague at Iowa State University. They have a very, very nice facility for doing vaccine and challenge studies. And in about 100 cattle, baby cows, we evaluated the safety and immunogenicity of the vaccine. We did that, sorry, I went back, and found in this first paper that the vaccine was able to reduce the clinical score in cattle, and as well as the body temperature, and the lesions you see here due to RSV, bovine RSV infection in cattle, are not seen in the vaccinated animals. So then we went ahead and continued this characterization of this vaccine according to toxicology under good laboratory practices, immunogenicity, also GLP, and then propose a human trial. But before doing the human trial, we thought maybe we could give a shot to this vaccine in a field study in cattle, because bovine RSV is a major problem for the meat and milk industry everywhere. So we did a field study here in south of Chile, in the border with Argentina, near in Patagonia, and we vaccinated several hundred cattle and tested the vaccine for immunogenicity and, of course, efficacy. And actually worked very well. So we found that one initial dose with a booster led to very strong immune response that was better than the current vaccine for RSV in cattle, and also is able to produce immunity against tuberculosis antigens. And tuberculosis is very important as a health problem in cattle. So we tried to push forward this vaccine also for veterinary use in the bovine industry. But since we complied with the FDA requirements, we went ahead and proposed a phase 1 trial that was conducted in 2018, I think, right before the pandemic. And this was done in healthy adults and tested for safety and immunogenicity. As shown here, we have 3 cohorts, and in none of them we saw any severe adverse event. We also only saw mild and moderate adverse events. And then we checked the production of antibodies. So you know, antibodies are able to neutralize viruses. This is an animation for SARS-CoV-2, but same applies for RSV. And we found that 2 weeks after the immunization in healthy adults, we saw significant levels of neutralizing antibodies against RSV. And also the immune response requires T cells that can recognize and destroy immune antigens. So usually your T cells circulate on the blood, and when they meet or they contact healthy cells, there's a process known as tolerance. So self-antigens are tolerated. But we will see that when the T cell sees an virus-infected cell, and this is a T cell specific for that viral antigen, which you induce with your vaccine, then this interaction leads to the activation of the T cell at the site of the contact with the infected cell, the cytokine production to bring in more cells of the immune system. But at the site of the infection, there is release of granules that contain perforin and enzyme that lead to the destruction of the infected cells by the creation of a pore on the membrane and the entrance of granzyme that promotes the apoptosis of the infected cell. And then we tested that in our study. I'm trying to move this a little bit to the side. Okay. And found, as I told you, antibodies against RSV and also anti-PPD. This is not Partido por la Democracia. No hay una declaración política. This is an antigen for mycobacteria. And we saw antibodies and T cells that recognize this antigen from mycobacteria. But most importantly, we saw antibodies against nucleoprotein antibodies and cells that recognize nucleoproteins from RSV. And we further characterized this, so we published several papers with this data, but this is a second paper showing the nature of the cytokines produced by these T cells in humans here in Chile, showing that they have the right profile for neutralizing viruses. So this is where we are at the moment. So we sort of discovered the vaccine, did many studies previous to the GMP. Then we did the GMP, more studies, several trials in different animal models. I showed you some mouse data and cow data. And then we went to the phase 1 and went well. We published this in Lancet. And now we are just implementing a clinical study in Europe under the European Medicine Agency regulation that approved this recently, and we are now implementing the sites. And Dr. Calvo, Dr. Juan Gonzalez, and Peñalosa are helping, because it's a tremendous work to do this in another country. All right, so we were going to move into that phase 2 trial, and then COVID-19 appeared, right? So this was December 2019, January 2020. So this was a major problem. Again, respiratory virus with a genome equivalent to RSV. It actually is able to lead to several symptoms, most of them dealing with the respiratory system. But pretty quickly we realized that other things were happening to people with COVID, right? So diarrhea, skin rashes, but this is what I'd like to focus on today, that has to do with the cognitive impairment of people with COVID. So we had already published 10 years before the pandemic a paper that I showed you just one result in mice and rats, showing that in learning tests, mice that were infected with RSV, and now we have a paper under review, the same thing with metapneumovirus. These viruses lead to learning disabilities. So we found the RNA from RSV on different areas of the brain. Also we can find antigen and protein. And when we did learning tests, this one is called Morris-Water maze. It's a learning test where you have a pool with water, and you set the animal on the water, and the pool has a hidden platform that is at the level of the water, so the animal swims around, and then if it learns, it's able to realize that that the platform is near the triangle and far from the circle. So the second time you do the assay, the animal knows has to swim towards the triangle and then away from the circle. So you can tell if the animal learned how to deal with this. So when we did this assay in animals that were infected with RSV, you see here in green the control uninfected animals. They take about 80 seconds the first time. The second time they take only about 20 seconds. But if the animal was infected with RSV, and it's very important to say here that this assay was done a month after infection, and the acute infection is only 3 days, 3-4 days in the animal. So these animals are not sick, they're not like respiratory, they don't have a respiratory disease at the moment to the assay. They show a significant deficiency in this assay compared to the uninfected animals. Then we did a second test. It's called the marble berry behavior. These animals live in cages. They have a bedding, and they hide everything under the bedding. If it's food, they hide the food from competitors, and the baby pups, the baby mice, they hide them from predators. So after half an hour, a mouse that is healthy will just hide all the marbles under the bedding. But a mouse that was infected with RSV, and again this was about a month after infection, has a significant deficiency at hiding the marbles. And we can restore the capacity of the animals to hide marbles when we vaccinate them with the RSV vaccine, suggesting that this is an inflammatory process. And this is actually shown here. So mock animals hide all the marbles. These are the RSV. But when we neutralize or block the entrance of cells, or the crossing of cells to the blood-brain barrier, we can restore the capacity of the animal slowly to respond in this test, suggesting that the blood-brain barrier had to do with this deficiency. And we proved that in different assays. So this is one of them using Evans blue, and we show that after infection, if you give the mouse Evans blue, then the brains become blue because the blood-brain barrier opens after infection leads to this entrance of Evans blue, and it's pretty impressive how these brains get this color due to a respiratory infection. But not only Evans blue is just an assay, but when we look for monocyte B cells and T cells, there is a significant increase of the number of these cells in the brains of animals that have been infected with RSV. And we published this paper in 2020. Now you can directly see if the virus can infect cells that are present in the nervous system. So these are astrocytes. This is a recombinant GFP RSV. You can see that after 24 hours, all the cells are green, suggesting that infection actually takes place in the cells. So all the data was derived from mice or rats, but this is the scary part. These are assays with infants. This based on our study published in mice and rats. Pediatrician and a psychologist, they did this study in lactating babies between 0 and 12 months, following 4 or 5 outbreaks of RSV in Santiago. And they found something that is very worrisome, because when they do these speech learning assays, and these are like sounds that you make to the babies and you show them like a little toy and they respond, and they use this little hat with electrodes so they can follow their brain responses. It's pretty impressive how here in light blue, the control babies that had either no RSV or mild RSV, they have this response when you make these sounds change, right? It's a phonetic trial. But if the baby had RSV, there was a significant lack of response compared to the controls. This is a pretty strong suggestion that the speech learning ability of babies can be impaired due to RSV infection. So the model, and we now in the last paper, we know this had to do with IL-6, which is a cytokine that promotes the opening of the blood-brain barrier. So you have infection of the airways, and some of these cells go back to circulation, and they carry probably like a Trojan horse model virus to the brain, and that leads to blood-brain barrier opening due to systemic inflammation. Now, when we think about COVID, going back to COVID, it's quite the same, right? So you have usually, for the most severe cases, hyperinflammation, and this also happened for Hanta. Paragines is here, it's a very typical thing, so very strong inflammatory response to a virus. For SARS-CoV-2 is the same, so you have this sort of cytokine storm that leads to severe disease. So if you want to vaccinate and clear the virus, then you need to produce neutralizing antibodies with a balanced production of cytokines. And of course, when we were about to initiate this phase 2 trial for the RSV vaccine, COVID-19 pandemic, the COVID-19 pandemic came up, and then at that time we were part of a network of international centers doing studies on vaccines, so we were invited to participate in pretty much all studies. But we wanted to go with this approach, with an inactivated virus approach, and with this recombinant BCG approach, because they have a few things in common. First, they are safe, they've been used for many decades as a platform. Second, you're going to have all the antigens of the virus. And third, this is a platform that could be useful for Chile, because the storage conditions for the vaccines was easy to handle. And so in addition to spike, so most of the vaccines that focus on only one antigen have focused on the spike. We thought that having envelope, membrane, and nucleocapsid was also important to induce a strong immune response. And then we published this paper very early, this was like April 2020, suggesting that countries that use BCG as a vaccine for TB, they have better responses to SARS-CoV-2 outbreaks compared to countries that do not use BCG. And that was in terms of number of cases and also mortality so countries that have BCG as a vaccine, they, for reasons that at that time were unknown, have better responses in the population. And now we know that had to do with trained immunity, that this vaccine is able to train the immune system and even in a non-specific manner respond to the virus. So we produced these recombinant BCG strains and we tested them in mice and showed and published this, that the vaccine was able to produce T cells leading to gamma interferon production, and we are now at the moment of moving this forward into a GMP labs. So the notion is that this cytokine storm, that is this exacerbated immune inflammatory response triggered by the virus, can be prevented by a combination of a vaccine that leads to trained immunity and also to adaptive immunity specific for the virus. We worked with many countries during the pandemic, the United States, Israel, China, and France. And we also established a consortium of universities here in Chile, and University of Austral was part of that. This is a formal agreement that we signed to carry out studies here in Chile for vaccines. We wanted to work on an inactivated vaccine, and there was one produced by that was under development by the US, another one, a couple from China, one from France, and there was another one in India. At the end, things moved faster with the collaboration with China, and the notion is simple. You take virus, large amount of viruses, you inactivate it with beta-propiolactone, and then they are mixed with alum as adjuvant, which is a very well-known adjuvant that's very safe. And in this paper in Science, they showed that the vaccine was immunogenic and efficacious in mice, rats, and macaques. So based on that, we proposed a clinical study here in Chile that showed that the vaccine was very safe. So the main adverse event was pain at the site of injection, and was equivalent to what we saw with the placebo, which also was an injection, right? The most important thing is that we found that the vaccine led to antibodies, neutralizing antibodies, 2 weeks after the second dose, and the frequency of zero conversion was over 85% overall, but 94% in the total vaccinated group. We were one of the few groups in the world doing T-cell assays, and this was in collaboration with Alessandro Sete, who's actually coming later today to give his show us his work. So they provided us with antigens from SARS-CoV-2 so we could evaluate CD4s and CD8 cells for the ability to respond after vaccination in these healthy adults. So we even were able to follow the memory response over time with different doses of the vaccine. I published several papers, over 20 papers we have published so far about this vaccine. So with this data, the vaccine was approved for emergency use in January 2021, and that led to part of the agreement that was to have 60 million of vaccines here in the country. At that point, we have received a few amount of vaccines from other places, clearly not enough to complete a vaccination campaign. And in January 2021, the vaccines landed in our airport in Santiago. Chile was up in the ranking of countries vaccinating the population. And of course, it was very exciting what we saw in the clinical trial, but the clinical trial is a control trial, so people are selected to be part of the trial. The inclusion and exclusion criteria is different when you vaccinate everybody. So that was a time very sort of like moving, exciting, but at the same time very complex, right? And we saw here in March already 2021 that the people that got the vaccine before, so elderly, mainly elderly, started to leave the ICUs, and then younger people started to go into the ICUs, because remember the vaccination process here was elderly people first and then younger people later. When the MOH, the Ministry of Health, they did an effectiveness study, they showed that the vaccine has an effectiveness of 67%, which is pretty high compared to the influenza vaccine, which is about 40%. This was very good to prevent symptomatic COVID, and of course it was better to prevent more severe cases of COVID-19. And you could see that the effect of the vaccine was the vaccination campaign was very strong. As soon as the campaign was initiated, there was a decline in the number of cases. This was our Delta wave, which was not very strong. And Omicron was high, but Omicron is why it was because the population was vaccinated, the number of fatal cases was reduced. And there was a constant decline in the number of death and ICU internalizations. In terms of safety, these are different platforms used for COVID. The inactivated vaccines, these are two inactivated vaccines, show some of the lowest adverse event frequency, which is now under discussion. About this strong discussion in the community about the adverse events that this vaccine could have. But there were some questions that we needed to answer. How long was the immunity, the need of additional boosting, neutralization of variants, and of course children, right? We thought that children were not susceptible to COVID, and that was clearly wrong. So we found that after 6 months after the second dose, there was a clear decline in the number of, in the titers of antibodies, so we have to communicate this to the authorities, so boosters were put in place, and that was pretty impressive because after a third dose of the vaccine, and even a fourth dose, we got these sustained levels of neutralizing antibodies that were stable over time. The same thing with T cells. There was not a very strong decline on number of T cells, but still having a third and a fourth dose led to more stable numbers of T cells in the circulations that were specific for SARS-CoV-2. And for the variant recognition for any vaccine, especially those that were focused only on spike, there was a clear reduction, a decrease in the capacity to neutralize the virus. And that was also the case for this vaccine, but this is very impressive. When we look at T cell immunity, we found that T cells that not only see spike, the neutralization capacity is focused on spike, but this is, when you look T cell immunity, you look for immunity to all the specific for all antigens. And you can see here very clearly that if you compare T cells recognizing the wild-type virus versus Omicron, there was no difference really in terms of their ability to recognize the antigen. So having this broad immunity led to a better response. And that was actually shown to be correct in population when they did boosting of the different groups. The effectiveness went up to 80%. Depending on the nature of the vaccination schedule. Now, as the vaccination process moved into the adult group, more and more countries started to report increase in the number of COVID-19 in younger kids. That was the case. This is Israel, this is the US. The same thing was happening in Chile. So we put together a trial because it was also very, very worrisome, this pediatric inflammatory multisystem syndrome in children. So we put together this clinical study that was larger actually, because it was from Antofagasta in the north all the way to Puerto Montt. And Valdivia was also part of this study. And we enrolled children and found that the vaccine was even safer in terms of adverse events than in adults. the immune response was better. We got like 4 times more antibodies and T cells in children than in adults. And also the ability of antibodies and T cells to recognize Omicron, that was the variant circulating at the time, was still pretty strong. That led to the rollout of a vaccination campaign in children in Chile that led to pretty impressive results. For example, in terms of safety, this is the frequency of adverse events in children, 0.01%. This is lower than any COVID-19 vaccine, but even lower compared to any vaccine that are given to children, like polio, measles, etc. So it's very, very safe. And in terms of protection, this was the effectiveness was greater, was over 70% in children, and this is the response of the cases of twins that were taking place in Chile. They were clearly down after the vaccination campaign. So what we did in Chile in terms of vaccinating, combining different vaccines was clearly, actually was able to work. And this is August 2025. This is official data from the Instituto de Salud Pública. And it's pretty impressive how SARS-CoV-2 is down here. It's the last virus. If you do the same study in the US, Israel, Europe, it's quite impressive that it's about 30% SARS-CoV-2. I was actually there a couple of weeks ago, and many people were sick. So SARS-CoV-2 is still circulating a lot in different places of the world, but in our territory in Chile, this is data from the official data from the regulatory agency showing that the vaccination process led to that this virus actually pushed to the last one circulating past winter. Now, we were sharing the data with different countries. So one of the things I want to say is that this data led to the approval of the vaccine in different countries like Brazil. Brazil has the largest pediatric population in America, and they have no vaccines for children, so this data led to the approval of this vaccine in Brazil. So what I told you is a combination between basic research and clinical research, interaction between the public and private sector, of course international collaborations, and, but the most important thing is that citizens, they need to understand what vaccines do, that they're safe. We need to be transparent in terms of telling, just explaining the results, good results, not so good results. It's important to be clear, because if you have a very good and safe and effective vaccine, but people use it, it's like no having a vaccine at all. I just want to thank many, many people, hundreds of people work on this, different universities, including University of Austral was very active, collaborating with us during the pandemic. And this is my team during the pandemic, so people were with masks in the labs, some in their homes, helping to analyze data. This is the first picture we took after the pandemic with no mask. This is the current lab. And this was a university during the pandemic, 2020, 2021, pretty sad, pretty empty, nobody was there, no students, no administrative people, no faculty. And this is the same spot, and herd immunity helps us to be together, like here today with no mask. So this was what I wanted to check. Thank you very much.
Any questions? I'm sorry. Pueden hacer la consulta en español, nosotros la repetimos.
Dr. Kaeki, thanks for the talk, it was very impressive. I have a few questions, and the first one regarding your RSV vaccine. That's very, very interesting, the model. How do you foresee if everything is going well and all the regulatory requirements are met and everything is working in a perfect way? How do you foresee the implementation of the vaccines, your model, because it's maybe the single model that I saw for this kind of vaccines and the current models of vaccines and novel antibodies for RSV in infants?
That's a great question. We have now to do the studies. I mean, to answer your question, the current studies are more challenging because we have immunization in progress, with immunization with a vaccine for pregnant women or antibodies. So that creates a challenge for the design of the study. But it's actually not a big issue for us because the Pfizer vaccine for pregnant women is designed to induce immunity against the F protein, and you get adoptive antibody production and then passive immunization of the baby. The same thing with the antibody, is passive immunization of the baby. So we think there's not going to be any kind of sort of cross-reaction between the passive immunization against F with our vaccine that leads to T cells against nucleoprotein, different antigen, different type of immune response. And actually we think they might be even considered to be used together, because if you promote strong T cell responses together with the strong passive antibody responses, so strong active T cell responses with the BCG vaccine together with a strong passive immune responses with the pregnant woman immunization, for example, then you get better immunity. So our goal is that the vaccine could be considered to be used on top of what is currently being used.
Great. And my other question is, do you see in the animal models any data that suggests the time of the length of the antibodies in the
protection?
Yeah, protection.
That's a great question. So we've been looking at this for over 180 days in mice. So it's a long time protection. But the most important thing is that we create T cells, memory T cells. And those are there forever, basically. Every time you get challenged with RSV, those T cells are going to be activated.
Great, thank you.
Thank you for the question.
Thanks, Dr. Calergis, for your presentation. Because I don't have too much opportunity to talk with you, I would like to know how are the plans, or how goes the GMP facility in the university, and also what is going on with the other vaccines that you are working in the lab, metapneumovirus, hantavirus, or other respiratory virus.
Right. So great question. Thank you, Dr. Varilla. And we have time to talk, so anytime we can meet anytime. We can go to Guzman and then talk with him. So the lab is built, okay, so, and the equipment is there, but complying with regulations in Chile is difficult, right? There's been a lot of discussion in terms of what we call the permisología. It's very complex. But we like to comply with it, so we're doing it 100% according to what is needed to comply with the regulatory agency. So right now, the only sort of the missing piece is completing the HVAC, which is the air conditioning and control pressure system. That's the last part. We left that to the end because you need to have everything in place when you like to set up the lab under motion, right? Because you need to have clean air, you need to have the correct pressure in every room, so that's what we do at home. So that's the situation. And now we like to make 4 vaccines, so RSV, metapneumovirus, SARS-CoV-2 and Hanta, all are BCG based. But also we have an antibody that we are planning to make GMP. And of course we will tell them that this facility will be open to other people and other groups in Chile and in other countries. And we have had people visiting from several universities in Chile. We invited them in January, Santiago. And also from other countries in Latin America and even from the US. We have 2 groups that are interested in eventually making a GMP antibody in the lab. But every product is different. So you need to get approval by the regulatory agency for each of those products. So you need to, it's a long process. So if you want to do something, we need to start talking soon, because it's a long regulatory process.
Another question.
Thanks for the presentation. My question is regarding the same topic of Mariana's asked. Do you think there is a path to get the approval of the vaccine, the RSV vaccine, without or bypassing the FDA regulatory approval? So do you think, my question is, do you think there is a path to approve the vaccine just with the regulatory agencies, Chilean agencies?
I think your question has an issue. In order to use the vaccine in Europe, no one cares about the FDA. If you want to have the vaccine approved in China, no one cares about the FDA or the EMA. And if you want to have your vaccine approved here in Chile, the only thing that matters is the Chilean regulatory agency. So that's the legal situation. But we have presented this vaccine, first we presented it to the FDA, and that was in 2017, 2018. And we got a very, very nice report from them. And it was so nice that we got this scientific suggestion to do the cattle study. So the cattle study that we did in Iowa, that was after discussing with the FDA a couple of times. You have meetings with them, you present the data, they give you feedback. It's actually pretty interesting. We scientists like that. We did the same thing with the EMA, the European Medical Agency, and we got the same situation, but we have everything for them. By then we have all the animal models, the cattle model, a phase 1 trial, and the EMA, the European, also approved the vaccine for clinical study. And here in Chile, we're dealing with the ISP, which has been changing a little bit, you know, about this probably. So we're still working with the Chilean agency. I have to tell you that the toughest one is not the FDA, and Alessandro's here, he can probably agree with that. It's not the EMA. The toughest one is the Chilean agency. It's like we're in Switzerland. It's really tough. And Mario knows about that, and Susan knows about it. So, but we like to comply with every agency that regulates what you do in the country. So at the moment we do a trial in Europe, which is approved by the EMA. We're trying to do a new trial in Chile, and it's been challenging dealing with the Chilean agency, even if we have approval in Europe. So that's the situation. But the issue to your question is that the only thing that matters is when you are doing the trials, or when you want, for example, Pfizer, they have vaccines approved, right, in different countries. Approval of the vaccine in Argentina doesn't mean that, or in the US, the FDA, doesn't mean that it will be approved in Chile automatically. The ISP has to review all the components. And you can, of course, provide approval from other agency as an element for consideration, but that doesn't mean that it will be approved. There have been many cases where you have a vaccine or other medicine approved in one country that was not, or by another agency that was not approved in Chile or in another country, for example.
Thank you for your talk, Dr. Kalergis. And it was really nice because you you talk about different topics of your research, right? I was wondering something about the neurological impairment effect of some viruses infection, because you showed some mice models that was really nice, but I'm not familiarized with the timing of those models. So these effects are seen, you see, more in an acute phase of the infection, or maybe it's for a long-term effect after the infection. Trying to think about this in human, where is going to be seen this effect when the people get infected or years after the infection?
That's a great question. The reason for doing this was I was just back from the US, 2004, here in Chile. And that year was a very, very strong RSV outbreak. Very strong, 2004. And there was a child that was a son of a friend of us that got RSV when he was probably like 3-4 months old. And he developed this very long-lasting sequela that was neurological. So that was, to me, was impressive because it was clearly due to the RSV infection. And at that moment, there was no study connecting RSV with neurological symptoms. But if you talk to pediatricians, they will tell you that about 3-4% of children that had RSV might have seizures or like clinical neurological symptoms, but those are pretty strong, right? When you have seizure and convulsions, that's pretty strong. So then we started this work and published the paper in 2012 in PNAS, showing that in mice and rats you can have these learning disabilities. We published the paper. I got so many emails from people all over the world, mostly mothers, saying that now I understand what happened to my kid, because they had an RSV case and they developed some kind of disorder. So based on those emails, I connected with a neurologist, and she did a retrospective study she had a cohort of kids with attention deficit disorder, about 400, pretty large here in Chile. And then she started calling all the parents, compared to 400 controls, and then she saw like 4 times more, a higher frequency of RSV or respiratory virus hospitalization in the cohort with attention deficit disorder. That was happening, in my group. But then I was showing this data in different RSV meetings. And then colleagues came to me saying, this happens in humans, but we don't think this is actually significant. That was for many years. Like we published the paper in 2012, and then the pandemic came in 2020. And then everybody was talking about these non-respiratory symptoms, cognitive symptoms. Because adults that have RSV, they can tell they are tired, fatigued, depressed, whatever. A baby doesn't tell, right? If it's fatigued, depressed, tired. The baby is sleeping all the time. So then the same faculty, they were telling me that this was not significant. After COVID, after they realized that long COVID was there, they started publishing papers showing the same for RSV in babies and in older adults. So this is real. And Dr. Falcone was telling me that in the past RSV meeting, I was in Rome a couple of weeks ago, I couldn't go. They focused on older adults because many adults, especially older adults, have RSV, and they develop dementia after pneumonia due to RSV. That's because the vaccine is so important. So this is all over. But the things that we're scientists, we try to be a little bit ahead, so we know the mechanism now that we're trying to publish that paper. We submitted that a couple of weeks ago. We think it is IL-6 that is produced in large amounts after RSV is aspirin to metapneumovirus infection, and that IL-6 leads to opening of the blood-brain barrier, and in some people that have susceptibility leads to cognitive disorders. That's the mechanism that we're putting forward. Thank you so much. Thank you, Dr.
Thank you, Dr. Kalakis. Now I will introduce to Dr. Alessandro Sete. He has had a long trip to be here. He was arriving to Sonno now in the morning. And he's professor and co-director of the Center for Infectious Disease and Vaccine Research in La Jolla Institute for Immunology in La Jolla, San Diego, California. He's adjunct professor of the Department of Medicine in the University of California in San Diego. His talk is Adaptive T-cell Response in Natural Infection and Neurodegenerative Diseases. Dr. Seth, please.
Thank you for having me. Sorry about the United Airlines got so much delayed. And it was very interesting to hear what Alexis was saying of the interaction between viral infections and neurodegeneration, and I look forward to talking to you about it. I'm not going to show that data today, but it's an area which is very, very important and very interesting. So I am going to start by giving you a little bit of a vignette of our studies in terms of SARS-CoV-2 in the landscape of today, in the landscape of a general population that has received multiple vaccinations and multiple infections, and so what is the fine detail there. And then I'm going to switch over completely to another set of data that we've been developing in recent times that has to do with neurodegeneration and in particular with ALS. So one important consideration when we go back to the SARS-CoV-2 pandemic is to highlight that there are very different roles of different types of adaptive immune responses. And it's important to distinguish the difference between protection against infection and protection against disease. And this is probably a message that could have been articulated more clearly during the pandemic, because a lot of people got confused and said, well, I got vaccinated 3 times and I still got infected, so what's wrong with that? The vaccines don't work. No, it's not that they don't work, but it's a little bit more complicated, a little bit more nuanced. So, in the context of protection against infection, really what you want are neutralizing antibodies. This is what makes okay. And in context of protecting against infection, adaptive cellular immunity has a relatively minor component. But the situation is quite different when you look at protection against severe disease, hospitalization and death. There, the role of cellular immunity is much more prevalent. And essentially, if you want to make a generalization, of course, the neutralizing antibody prevents the infection from taking place. But once the infection has occurred, we really need a T-cell response to eliminate the infected cells that otherwise would continue to support the infection and cause the disease. So what is the evidence that speaks for a substantial contribution of T cells in the context of SARS-2 immunity? Early on in the pandemic, we highlighted the fact that if you look at early after infection, the best predictor of having a mild disease is having an early, strong CD4 T cell response. That is the best correlate by far. Also, an early CD8 response correlates Well, the antibody response, the early antibody response actually has no correlation at all with the disease trajectory. This was also underlined by a number of studies. The first study was actually from Bertoletti's group in Singapore that showed that an early T-cell response also correlated with shorter duration of viral titers and lower amounts of virus being produced, which obviously makes sense as an impact on a milder disease. And we also reproduced that data in a much larger study in 2024. Also, you might recall that Italy was hit very hard and early in the SARS-CoV-2 pandemic, and we had a number of collaborations with the Italian groups. I'm Italian, so I have a lot of connections, and so we got samples from them, and in that study from 2022, when we finally analyzed all the data, we saw the same thing, but there was a correlation between early T-cell responses and mild disease outcome. But also, importantly, having a T-cell response that targeted not only spike, but also nucleocapsid and other non-structural proteins was also a correlate of milder infection. So, as I was saying, we became interested in understanding the interplay between infection and vaccination. And in the start of this study, we had a large cohort of people that we were following. Actually, in the start, we had two cohorts. We had a cohort of vaccinated people, and then everybody that got vaccinated got infected. And then we had a cohort of infected people, and everybody that was infected got vaccinated. So at the end of the day, became a single cohort. And this cohort we've been following now for a number of years, from 2020, so we're in year 6, and we will continue following them. And actually, it's an interesting cohort in our mind because it does show the interplay of infection and also for flu, for RSV, for whatever. We have a 10-year of periodic blood draws and serum. You can really see the interplay between different infection and vaccinations. So, but we were curious about very basic questions, as is what happens when you boost with mRNA vaccines a number of different times. Do you get to a plateau, or you continue to get a benefit, and what does the repeat vaccination do to the durability of responses and so forth? So at the same time we were doing this, we developed a T-cell assay that we followed and still use to get insight in SARS-CoV-2 infection and vaccination history. And the assay was established and validated very early on with samples from different cohorts, people who were only vaccinated, people vaccinated and infected, people infected, or people that were not exposed. And the assay is very simple conceptually. You do a T-cell assay and you plot on one dimension the T-cell response against spike, and on the other dimension you plot the T-cell response against everything but spike. And so people that have vaccinated and are not infected are the blue people. They only see spike. While people that have been vaccinated and infected obviously see both spike and non-spike antigens. Interesting, you see here that people who are infected compared to people who are infected and vaccinated, the people who are infected and vaccinated have a higher component of spike because they've seen spike twice as opposed to only once people are only infected. So this is a fairly reliable indicator that someone was vaccinated or infected. You can discriminate. So in this cohort, we had the people, we're collecting the samples, and of course, as we were going through 2022, here is plotted to 2023, at the same time we were collecting these samples, there were all sorts of waves of variants coming and going, and these people that we were following, we were asking have you experienced a SARS-CoV-2 infection? And they all said, no. So we were specifically monitoring the uninfected, and they all said that they had not experienced respiratory symptoms and they had not been scoring positive in a SARS-CoV-2 test. Well, not really true in the sense that actually when you look at the T-cell responses of these people, we can see that clearly at least a third of these people had experienced asymptomatic infection. So they didn't know that they had been infected. Right? Clearly they had been infected. They had very high T-cell responses against non-spike antigens. So how does the T-cell and antibody response look in this situation? You see the antibody response here gets the first immunization, goes up, then goes down over the course of 6 months or so. We get another booster, goes up again, down again, up again. So essentially you see that after 3 or 4 boosts, the antibody response stays in plateau, at least in the course of 6 months to 8 years. And the T-cell response actually, even after the first vaccination, goes up and then stays. It's fairly durable, a lot more durable than antibody responses, which fits with what we know. We were interested to see if there was a difference in quality of the T-cell response. So we did a lot of single-cell sequencing, isolating the antigen-specific T-cells. And this is the bottom line, what we saw. A lot of different T-cell subsets that mediate the spike-specific antibody response in these individuals. These are all people that are either not infected or asymptomatic, because we excluded from analysis anybody that had a symptomatic infection. And you see the usual T-cell subset, Th17-like, Th2, Th1, Th8, and so forth. But also, very strikingly, we saw a very large component of Tregs that were detected in the antigen-specific response. The CD8 compartment was also characterized by a large, relatively large number of different subsets, and the most prominent was the granzyme high. So these are the classic CD8 killer cells, which were prominent in the study. So one of the questions that we wanted to address was whether the repeated vaccination was bad. At that point in time, there was a lot of debate that, like, oh my God, you're going to vaccinate too much, and it's going to lead to exhaustion, and we see absolutely no evidence of that. People that have been and continue to be vaccinated multiple times, they don't have any sign of T-cell exhaustion by either looking at a signature of mRNA expression associated classically with exhaustion, either looking at whether these cells continue to make cytokines as an expression of functionality, and so forth. This is both for the CD8 and CD4 response. And now, finally, the last piece of data in this context was going back to the this Treg component that we saw being associated with this particular cohort. And basically, what the data here shows is that in the cohort, again, there is no sign of exhaustion, and the cells, the T cells continue to be fully functional, and but there is this Treg population, and this Treg population is specifically associated with people that have had this asymptomatic, actually now we know also asymptomatic and symptomatic infection. And so we think that this is a good thing, an adaptation, in the sense that as you get productive infection, you still have an immune system that can fight the infection, the T cells are fully functional, but also at the same time you develop a Treg population that is helpful in preventing tissue damage, and that's why you get an asymptomatic infection as opposed to a more symptomatic infection. And so this is where we stand, I think, with a SARS-CoV-2 infection and disease, where basically we are now in a situation similar to what you have in influenza, where you have a population that has been heavily exposed to both vaccination and infection, and so you have a dual component of an immunity wall that protects from more severe disease. Still, infections occur, but the burden of disease is nowhere near what was during the pandemic. And I think we're seeing more and more that really asymptomatic infections are frequently in the case of SARS-CoV-2, but are very frequent also in other contexts. So I'm not going to show you today the data, but we've been seeing the same thing in the context of Bordetella pertussis, in the context of group A strep, where once you define the appropriate antigen, you can actually see that there is a lot of asymptomatic infection, and these asymptomatic infections are very often associated with the development of this Treg population, which I think is a powerful adaptive mechanism, so that upon reinfection, the pathology associated is reduced, while the immune system still does its job in terms of clearing the infection. So, and these are the various people that have been involved in these studies, key contribution from Alba Grifoni and Ricardo da Silva, and many other people in the group. Now let me shift to, instead, ALS. And the key points that I'm going to talk to you about is the fact that in these studies that were published in October of last year, we identified for the first time a particular protein as autoimmune targets associated with ALS. C9orf72 is a protein that, when mutated, is associated with much increased frequency of ALS, but this recognition, autoimmune recognition of C9orf is true whether people have mutated or wild-type C9orf. The other striking result of this study was that in the same person, we see both an inflammatory and an anti-inflammatory response against this C9orf protein. So there's kind of a battle of two different immune responses going on. And strikingly, the people that have more anti-inflammatory response are projected to live longer. So obviously, this is also interesting because it doesn't take a rocket scientist to imagine that if you could shift the balance more in favor of an anti-inflammatory response, that could have a therapeutic benefit. So what is ALS? You may be or may not be familiar with this disease. It's a very bad disease. It's fast. In about a couple of years, 3 years, usually the person affected dies, and it's also a painful death, because people essentially lose their capacity to operate their muscles and eventually suffocate because they can breathe. And there is absolutely nothing that is available to these patients. So, ALS is a neurodegenerative disease that is basically attacking the motor neurons, and it's associated with proteins that are linked to aggregation and alteration of lysosomal pathways and phagocytic function and so forth. And specifically, there are here 4 of the main proteins that are classically been associated with this disease. So we were wondering whether these proteins that are associated with disease, in the sense that mutations of these proteins is associated with increased incidence of ALS, would also be potentially recognized by an autoimmune reactivity. And so we synthesized overlapping peptides spanning throughout the sequence of these different proteins, and then asked whether PBNC from either healthy controls or ALS patients would recognize these peptides after a 2-week in vitro expansion culture. So the 2-week in vitro culture was introduced because in our experience, when you go into an autoimmune scenario, the cells that are specific for autoimmune antigens are usually found in very low frequencies, so you need to expand them. Otherwise, if you go with an ex vivo assay, you won't see very much. And at the end of the 2-week restimulation, we measure a number of different cytokines. So, for example, interferon gamma as a prototype pro-inflammatory cytokine, and IL-10 as a prototype anti-inflammatory, and IL-5 is often also anti-inflammatory. And basically, to cut the story short, we saw that CNRF72 was the one protein that was significantly recognized in higher, much higher frequency in ALS people. And also interestingly, the difference was most pronounced when you looked at IL-10 or IL-5. Interferon gamma was also significant, but really there was a strong anti-inflammatory component. We further investigated the type of response that we were seeing after the 2-week expansion, and we saw that in those expanded culture, CD4 T cells were the predominant T cell type. And when you looked at what cell type was the one that was making, for example, interferon gamma or the other cytokine, was overwhelmingly a response that was functionally originating from the CD4 T cells. And this is perhaps not surprising by the fact that we were using 15-mer peptide for expanding the culture, but still was clearly a CD4 T cell response. So, in conclusion, as I was saying, the CNRF72 is an autoantigen in ALS, and it's a CD4-mediated response, and skewing of a response towards a Th2 Treg phenotype. And the other points are also discussed in the paper. I'm not going to go through them in the interest of time. We mapped the T-cell response in the various different ALS patients to see which particular peptide were recognized. And as you can see here, the response is really spread out throughout the sequence of the protein. And so there is not a single particular peptide, and it's a very broad, multi-HLA specific. In fact, the peptides that we see are most dominantly recognized. The peptides that we see are capable of binding multiple HLA types. And we looked at different isolates, different biological correlates in terms of, for example, age, sex, different things. There was nothing particularly striking in terms of the correlates with age or sex. Then this other thing at the on the top is correlation with the ALS-FRS-R score, which is basically a score of disease severity. It's a composite score that indicates whether the person is capable of living and feeding and dressing themselves. So it's how impacted the life is, and other functional characteristics. And so there was not really a strong correlation. There was maybe a trend in some cases, but the P was not significant. So there is not a strong correlation between the T-cell response and, per se, the disease severity. But as I was saying, what was striking is that regardless of the disease severity, what was the best predictor of life expectancy, whether it was short or long, was the IL-10 production. And so that was the main result. So this says that you can have a disease that may be influenced by the strength of a response or whatever in terms of the severity, but really the disease progression, how rapidly you go down, is influenced by how little of a regulatory response you have. And in fact, that there are also precedent for this. There is a group in Houston led by Stan Appel that has done clinical trials. This is in a non-antigen specific way, but they've given low-dose IL-2 and CTLA-4 IG in people with ALS, and they observed some benefit in terms, again, of reducing the disease progression. So really the concept is intriguing, but increasing the IL-10 Treg function in these people may have some significant effect. So now let's go back for a second to the mutation. I remember that I mentioned the fact that C9orf72 mutations are associated with very high risk of developing ALS. Actually, by the age, I believe by age 55, 80% of the people that have this mutation have died from ALS. So it's a really high penetrance mutation. Interestingly, the mutation is in the non-coding region of the protein. So the sequence of the protein is absolutely identical in people who are having a mutation or not, which is peculiar. What the mutation does is it is in this non-coding region, and the mutation originates this runoff of denucleotides, crazy long, and so these denucleotides then get translated in dipeptide aggregates. And so one of the tempting hypotheses is that this dipeptide also contribute to damaging the functioning of the lysosomes and create aggregates that then contribute to damaging the nerve cells. But we were interested in asking the question, as I was mentioning in the start, we were seeing this higher C9orf72 reactivity both in people that had this mutation or not. But we asked, is there a difference if we compare people with a mutation and non-mutation? And basically, to cut the story short, again, the people that have the C9orf mutation have a much higher reactivity against C9orf, consistent with maybe forming aggregates with lots of dipeptide and so forth. But this opens another, at least for me, very interesting question. What about the people that have a mutation but don't have ALS? As I was mentioning, people that have this mutation, 80% are dead by the time they're 55. But if you look at them before 55, or the people with 20% that had not succumbed with disease. What's the situation there? And the reason why I'm very interested in this thing is of the previous studies that we have made in the context of Parkinson's. Now, Parkinson's, you may know, of course, the hallmark symptoms of Parkinson's are cognitive defect and tremor and shaking and so forth. But before you get those symptoms, there is a long prodromal phase of 10 to 20 years where essentially people are destroying the substantia nigra, which is the part of the brain that is whose destruction is responsible for Parkinson. But they don't have the symptoms yet. And this is, in a way, if you want to think about it like diabetes. Diabetes means you can't make enough insulin, but in autoimmune disease there is a long process by which the person is slowly destroying the islet cells that make insulin, but still enough is made so you don't get the onset of disease. But when you go below a certain threshold, then you have diabetes onset. So we are curious to see what's going on. Is this T-cell reactivity against C9orf preceding the onset of disease, which goes to the fundamental question, which is a question in all of human autoimmunity, whether the T-cell reactivity is the chicken or the egg. So do you have a T-cell response because there has been tissue damage, or there is a T-cell response that causes the tissue damage? And of course, it doesn't need to be a clean one or the other, but it certainly is important to understand that. And also, if this reactivity would be preceding the onset of the damage and the clinical disease, again, it opens the hypothesis that you could, especially in people that have the mutation, you could intervene before the disease onset and augment IL-10 or polarize or whatever. So in closure, this is the model that we have of an amplified damage that in the neurons that results in aggregates and activation of CD4 and CD8 response, and conversely a Treg response that can inhibit inflammation. And I like to acknowledge the many people that have contributed to these studies, both in my lab and the many clinical sites that have been generous, and the patients that have been generous in donating PBNCs for us to perform these studies. Thank you very much.
Thank you, Dr. Seta. Right, Doc? Any question?
Hello, Sandro. Thank you for making it so far to Valdivia.
Thank you for having me here.
Despite all the delays. So I have some questions regarding well, first of all, I'm very glad that your group analyzed the Tregs in the context of vaccination of SARS-CoV-2 and natural infection. I was so surprised that for, I would say, at least 2 years, nobody would look at Tregs for SARS-CoV-2. It took a while until people went to it. So at least there's data for that, and I was sure that they were obviously going to contribute to attenuate disease. So I'm glad that data's out. Regarding the autoimmune disease follow-up you're carrying out, so do you know already the HLA that's also associated with the presentation of those peptides in such a way that maybe you could now go into looking for those T cells with the equivalent of tetramers, polymers that exist nowadays for that?
Yes, excellent question. The tetramers, I don't know, but it's my bet is that the signal is not going to be sufficient.
Dextromers now, I think they're called.
All right, but it's still, the HLA restriction is very much at the center of what we're interested in for a couple of other additional reasons. So, A is not an HLA-associated disease, just epidemiologically, and we don't see a particular HLA being more dominant. So we see a broad spectrum of HLAs. The other point is that we're interested in HLAs and cloning and determining the TCRs associated with the disease, because we think that maybe one way to approach this practically is to make Treg transfected with specific TCRs. So it's a CAR T, essentially, approach, but with TCRs. And why to do a TCR-T as opposed to a regular CAR T, which would be simpler? Because C9orf is intracellular, so to have an antibody against C9orf in a CAR T setting would not be expected to do much. So we are drilling down at the TCR and HLA level, because I think that maybe this is it's a path forward.
So maybe in that line of the Tregs, what's your take or impression on the fact that we have to think on CAR T cells for Tregs, and we don't have any good adjuvants or anti-adjuvants for producing good Tregs based on immunization of a peptide for which we want.
Yeah, so I'm not the best adjuvant guy, but certainly even in a broader context, certainly if you could reliably induce antigen-specific T cells in vivo, that would be great. I'm not sure that we're there yet, at least that I know of.
I think we've never been there. I don't know why. We have never looked for adjuvants that induce Tregs for autoimmunity.
Yeah, I don't know. But also, along the same lines, the other approach that would be easier would be to just take the Tregs or take T cells out of these patients and grow Tregs out of them or expand them and then put them back, right? In fact, there is a number of companies and groups that have done that. And the problem I hear, I haven't seen the data because it's not published, but what I hear is that one of the main problems is that once you put the Treg back in the patients, they revert to having an inflammatory phenotype, so that actually it's a big concern, because you can end up killing the patient if you give them a big dose of an autoreactive T cell. So I'm with you. I think it's a very interesting line of investigations, and there's a lot that needs to be done still.
So, last question, maybe there's no... So it's true that the Tregs can revert to interchange with Th17. They have like a common program, so it's very tricky. My last question is whether, when you saw the Treg assessment, the measurements of Tregs that tells you that those that have higher Tregs are better in prognosis, do you have that in time? It's like not just one time point, but different time points, just if it varies?
No. And actually also I want to point out another limitation of our studies is that as I said, the measures are done after a 2-week expansion, and so a 2-week expansion can totally screw up the phenotypes. So I call them Tregs. I really don't know that they're Tregs. I know that they make IL-10. The question is, do I see these changes longitudinally? No, we don't have longitudinal cohort, and part of the problem is that the disease is quick. So people that are newly diagnosed, I mean, it takes a little, so you need to reach for people, right? So you don't get someone that has been diagnosed yesterday. Usually they are, and then quickly become so incapacitated that they will not come to you, or I mean, they're bedridden. So it's conceptually yes, but it's practically difficult.
Thank you very much.
Yes.
Thank you for your talk. It was very nice. I just had a quick question about the first part of your talk in terms of the unexposed individuals with a regulatory T cell response to the SARS-CoV-2. You had said that they're targeting mostly non-spike antigens. or is that correct?
Keep going.
Okay, yeah. So at least they just had a wider breadth of response to SARS-CoV-2 in general. I was wondering, and what you thought about the potential involvement of those responses being involved from seasonal coronaviruses, so previous exposures to, or cross-reactive exposures, which may or may not actually elicit higher levels of regulatory cells from previous infections. So could you kind of just talk?
Okay, so many good points from your question. So first of all, the recognition of non-spike is a hallmark of infection, whether it's symptomatic or non-symptomatic. It's just because for us it's easy to tell that there's been an infection if all of a sudden you start to see non-spike, because otherwise the spike, given the fact that everybody is vaccinated, but back then you couldn't tell the difference. So that's one thing. In terms of the reagents we use, we have, it's not perfect, but we're taking care of eliminating peptides that were cross-reactive with common cold coronavirus. And so we believe it's mostly, if not only, a SARS-CoV-2 response. But certainly pre-exposure to common cold is generating a response that is cross-reactive. But eventually, the regulatory response that is associated with symptomatic or asymptomatic infection, it is directed against all sorts of things. In the data that I showed you, those were cells that were sorted for responding to spike. So we're mostly spike, but actually as we go along, and also we see this now Treg expanding, many of them may actually be bystander cells. So that one potential scenario is that when you get an infection, then that activates also Tregs that are expanded, and some are antigen-specific Tregs, and some are just regular Tregs, because your body is trying to say, okay, we have an infection, the CD8 T cells and the antibodies are on the way to clear infection. Let's try not to destroy our lungs in the process. And so you have also a orchestrated compensatory phenomenon. That is my speculation.
Thank you.
Sorry. Thank you so much for your talk. My question is regarding the autoreactive T cells that you showed us. And I think this is going to your last point question, where whether it was the chicken or the egg. But since these are mainly CD4 positive, and considering the IL-10 factor in all of this, do you think that these autoreactive T cells were initially maybe part of the protective immune system and then became dysregulator in this maybe whole inflammatory environment, or something set up this to dysregulated phenotype instead of maybe protective phenotype?
You're talking about the first part?
Yeah, the ALS autoreactive T cells.
Oh, the ALS?
Yeah.
So I don't think that a interferon gamma or IL-17 response would be protective, right? Because they are inflammatory, and then that it's a self-antigen.
No, but I mean, when you see that these autoreactive cells are mainly CD4 T cells,
right?
Do you think that this may, at some points, directed to protective response, not mainly anti-inflammatory, but then they become dysregulated given the response that you see on the disease onset?
Well, those are all antigen specific.
Yeah.
Right. So I I'm not sure I understand the. So you're saying that they were specific for some other antigen and then they become.
I mean, we're thinking about Tregs. Tregs supposed to maintain the tolerance. So these are antigens that are on the body. So what I'm thinking right here is that maybe these Tregs were in circulatory system and then they became dysregulated. And they can be part of this onset of these problems, mainly in this case ALS, but maybe in other pathologies.
I don't know. The fact that we see that if the more of these IL-10 producing cells you have, the longer you live, would tend to suggest that they are not dysregulatory and dysfunctional in the sense of causing disease. It is possible that there might be a biphasic thing which would go back to being addressed if we are able to get samples from people that have the mutation but don't have a disease. What does the T cell response look like? That would be an interesting question and probably would also to some degree go and address what you were asking me.
Thank you.
Thank you, Dr. Seta. Next questions will be in the coffee, because we have the coffee now.
Yes.
I will introduce me by myself. After my talk, I have to go for some time, then I will be back in the afternoon. Well, my name is Mario Calvo. I'm a physician, I'm an infectious disease specialist and internal medicine specialist, and I'm professor here in University of Chile. Well, I will talk about clinical manifestations and immune response associated to COVID-19 infection in Chilean patients. But I would like to subtitle my presentation as how my scientist friends helped me to realize why my COVID-19 patients were the way they were. Well, when we saw COVID-19, we told us this is a different disease. This is not as before. This is not a sepsis, it's not a pneumonia, this is not an influenza. Which were the surprises? Younger patients had very mild disease, but they had PCR positive, and sometimes they were negative for serology. It was a surprise. What else? Sicker patients had higher levels of antibodies. It is not acceptable. The doctors knew that antibodies were good. Why they had so much antibodies? And there was a big difference among the severity of different patients. And we have several special patients as hematologic patients who maintained a positive PCR for COVID during weeks, and the severity was increasing slowly, and we didn't know how to treat them. Most hospitalized patients were rapidly worsening, and after a plateau, they were improving fastly. That was great. Normally, severe disease are going up and going down. Patients are not so clearly going up and down. And we have a bad news when we saw that after a successful discharge, some patients were going back with pulmonary embolism. All of that was what is happening here. To answer this question, my friends were in a big collaboration. The projects were written by María Inés Barría and José Luis Garrido, they are here, including their labs and teams. In Valdivia, we have the infectious disease team, hematology teams, and blood bank personnel. And we have the collaboration also from Puerto Montt, Temuco, Talca, Concepción, and Chillán. And there was a group of New York with a cohort from there, but the investigator who analyzed the data was Dr. Garrido, Chile. And also, all volunteers were Chilean. We had several cohorts. The first convalescent cohort was from Valdivia and Osorno. It was composed from 70 ambulatory convalescents. 36 were recruited from an outbreak of healthcare workers in Osorno, and 47 were blood donors. Blood donors for research too about treating with plasma patients with COVID-19 in the hospital Valdivia. We used, and this is so important to understand the studies, a score that was developed by us about symptoms and more severe, more symptoms or more systems were involved in the disease. The second convalescent cohort was a small cohort, 28 patients, and they were compared with 29 before the pandemic, and severity was also based in symptoms. You must remember that at that time, it was the early phase of the pandemic. We didn't have any universal score to compare with other research. And the third cohort was a prospective cohort of 59 patients that was recruited in different cities of Chile. That cohort included ambulatory and hospitalized patients. 16 were ventilated. We needed for the research of Medina to choose eight selected patients for severe and for mild for transitional change study. It was difficult, and I was menaced by Maria Ines, who told me that you must do it well because it is so expensive. But we did. Well, the third cohort was clearly different between mild and severe because mild patients were younger. But the most important difference is here about the comorbidities. You can see severe patients are related to several comorbidities, and this is the classical picture of a severe patient with COVID-19. We used here the score NEWS2. It is well designed for severe patients because it includes respiratory failure and hemodynamic failure. I want to take a minute to make an acknowledgement to the health personnel who helped us to perform this research. During that time, I remembered so many times this film about the people who transported nitroglycerin during the oil extraction. Because at that time, we didn't have any vaccine, and really it was difficult to go to the patient's house to get the samples. We traveled in car to patient's home. All the windows were open because we didn't know if any person from the team had acquired the disease. We have isolation techniques, but they were difficult to apply beside the car before entering the house. And finally, the major challenge was to sign the informed consent because you needed to take your pen, to take off your glove, to give the pen, to take the paper again, and finally we kept that in bags during months. And really the last ones were opened this year in the summer. Well, the questions. PCR positive but negative serology. We had patients, all of us, with less symptoms or asymptomatic, and for example, in your cohort, there were 2 patients with PCR positive. We were worried because we didn't know if people imagined the symptoms at that time, and we knew that anti-spike titers were normally measured by ELISA. And Dr. Garrido quantified the levels of anti-spike IgG with endothelial cells transfected to express the SARS-CoV-2 spike. And he demonstrated that those patients with PCR positive, they had antibodies against spike IgG. Then they had COVID-19. That was the proof. Moreover, he demonstrated that there was a relation between the anti-spike IgG titers and the severity of the disease. But, surprise, most antibodies and more severity. It is contradictory, it seems to be. Well, that's the question too. We know that normally first neutralizing responses are mediated by IgA and IgM. But we can see that it's correct that higher IgM titers and higher IgA titers are related in COVID-19 with more neutralizing response. But what did Fuentes find? Patients with lower and higher severity score are not related with the level of IgG titers, anti-spike or RBD, nor neutralizing response. But differences are evident when analyzing their NK effector functions. We can see that here. Humoral immunity are not related with the severity of disease. We have different type of antibodies here, but cellular immunity, you can see NK response are higher in the mild patients. Then we have a difference with some information of other disease. We can see that here too, where NK cell response are higher in patients with mild symptoms, but antibodies are related to more severe symptoms. We can see that here too, when the higher antibodies are presented in ICU patients. Then, there could be a second explanation about this that is related with neutralizing activity. We know that most of the neutralizing activity for SARS-CoV-2 is concentrated in the RBD segment in S1, about 90%. Nonetheless, there are antibodies against S2 segment in some people, in the general population. That means that beta coronavirus, the seasonal OC43, are related to this immunity. Could it be related really? Yes. We can think that early in the pandemic it was reported that this type of antibodies against OC43 could be high after SARS-CoV-2 infection. And Garrido studied the antibodies against same regions of S2 segments, and he described several associated with a milder infection and some of them associated with the severe infection. We can see here HR2 associated with mild disease and S2' FP associated with severe disease. Then he published this ratio between antibodies against HR2 over S2' FP, and you can see that it's clearly higher in mild patients. Then OC43 seems to be an original sin to the immune response against SARS-CoV-2. Well, anyone can say that it could be the virus that is changing among patients, but it is not really true, because at the early phase of pandemics there was not heterogeneity of viruses as now. We have several variants, but at that time we didn't have. The difference is in immune response. In the third cohort, COSAC studied that in mild patients there are several genes that are downregulated or upregulated. It's very interesting those that are downregulated because they include toll-like receptor, pro-inflammatory signalings, and complement activation. All of them we see in intensive care in patients with sepsis. They talk us about inflammation and severe disease. But we can see that mild patients can control this response, and we have a mild disease. Finally, the big difference in severity is a different phenotype. In mild patients, we can find NK cell response associated with good regulation for Treg cells and antibodies that stimulate NK cells. By contrast, in severe disease, we can find high neutralization with low response of NK cells and exhausted Tregs that produce a severe inflammation and damage to the lungs. Fourth question, hematologic patient. What happens with hematologic patients? It was difficult to answer well this question because we saw many patients with lymphoma, especially in our center, and also in some other centers that have the problem that this patient maintained a positive PCR for SARS-CoV-2 with a low CT, that is a high viral load. But the response could be in the Treg cells. You can see that lymphocytes are damage in lymphoma, and you can see that T results are higher during the first 3 weeks in mild patients. Possibly, they are so important to eradicate the virus. Then, it could be shown in other models. For example, T-Rex response are diminished to, T-Rex number is diminished to with increasing age. That's an explanation why older patients are so severe. And we know that lymphopenia was described early as a risk factor of lethality for SARS-CoV-2 infection, and also we can see that it was described by other groups as a risk factor for mortality in cancer patients. Then lymphocytes are clearly important in our response. 5. What happened about the recovery of this patient? Well, here Medina did a transcriptomic analysis comparing 4 and 4 patients, mild and severe, and I will talk a little about the MARN. The mRNA was measured in PBMC, where he looked for mRNA and fragmented, and after it was sequenced to describe the genes that are expressed in PBMC. Then we can see in this data that response associated with NK cell cap genes over time are present in day 0 and 7 in mild patients, but they are not present in several severe patients. But you can see that after 28 days from date of onset of disease, their representation of NK cell cap genes is similar between the two groups. Then we can see there is a delayed response in patients with severe disease. It's possible this explains why patients are worsening, but when the cellular response appears, they improve. It could be more well described in this graphic that is in the same paper, that is a protein-protein interaction network for the NK cap genes. And in this network, there are some proteins that are more important. We can see, we can see these proteins, and you can see the same. During the time of acute disease, the first week, we have a high level of expression of these proteins from NK cap cells, cap genes, in mild patients. But after that time, the levels converge between mild and severe patients. Last question. The problem with the thromboembolism. This was a headache, really. Well, during the research in transcriptional genes, we saw a module that is the brown. This brown module contains 508 genes. Those genes are related with coagulation, and the most important and significant function associated in this research is platelet activation. Those genes are associated with integrins, platelet glycoproteins, and von factor. All of them coagulation functions. Then, after that, I knew that all my patients had to receive after discharge anticoagulation for some weeks. As conclusions, we can see that titers disease, they are not related to a good response when we are talking about acute disease. It is different than vaccination, that's another reality. But the effector functions rules. Clearly, cellular immunity helps to these patients, but we can see that severe patient has a regulatory failure in which inflammation persists over the regulation of T cells. Okay, that's all. Thank you.
So thank you very much, Dr. Calvo, for your talk. So it's open for questions. Alex.
I love the regulatory correlation. Which markers were you using to define the Tregs?
I will try to ask for the help of Camila Cossack. She is here and she did the experiments. Thank you, Camila.
Yeah. Well, we use we selected the CD4 positive T cells, so it's CD4 positive, they are memory T cells, and we use CD25 positive, CD127 negative cells as whole Treg population, and we also saw within this population the FOXP3 population. And we saw the main difference in this bigger population, the CD25, CD127 difference. We also saw the PD-1 and the CTLA-4 expression in these cells, and we see some difference, but they were just trend on our cohort. And but yeah, the main phenotype of Tregs in mild patients were mostly regulatory T cells, instead in comparison with the severe cohort, which showed a less suppressive phenotype. That's the main conclusion of that paper.
Thank you very much. More questions? From this side? Mario, I was wondering regarding the other kind of experience you had with, for instance, with the response to immunization and also for COVID-19 for HIV positive patients?
Yes, I don't have too much information about the HIV patients. We know that the HIV patients were described as a risk factor population, but it was not so severe. But I believe it was caused because they were treated patients with good levels of CD4. Yes, I remember we had only one patient hospitalized.
Okay.
I believe the T cell functions were preserved in those patients.
Perfect. Even for without vaccination?
No, no, they were they were closed in their homes during too much time, and they almost all got the disease after vaccination.
Okay.
Almost all of them, yes.
Well, that's a risk population.
Yes.
They were vaccinated.
Yes.
Yeah. Okay. Thank you very much.
Yes.
More questions?
No, I believe the most important in HIV patients, most of them are younger.
I see.
Yes.
More questions? Maria.
Thank you, Mario. I was wondering about your experience with the plasma treatment.
Yes, yes. Really, if you see the research, it was a failure. But after we have some, it was a great problem to decide if it functioned or not, because we have several papers that talk that it's good and others it is not good. But I believe if you see the papers when it functions, always the administration is so early during the course of disease. And I'm convinced from two patients, microcystic, that they received the plasma very fast. And they have a very good evolution in spite of they were developing pneumonia. And the evolution was broad after that. It was broken really. But if we published the paper and it was a failure for us, if you see all the group. Yes, and the same experience had Elvira Balcers in the Catholic University.
Thank you very much. More questions? Okay. Yeah, one more question. Alex.
Still on the regulatory cells, I wonder whether you had a chance to look in other respiratory pathogens. So is it a general thing that disease severity inversely correlated to Tregs if you look at RSV or flu?
I'm trying to look into that as well.
Yeah. We also are performing single studies right now to deep further into different respiratory diseases about that topic. And I mean, of course, some more open questions, topics related to different respiratory virus infection. But yeah, we're focusing on that too.
So thank you very much, Dr. Calvo.
Thank you. I will introduce to Dr. Susan Bueno. She is full professor from the Facultad de Ciencias Biológicas, Pontificia Universidad Católica de Chile, and she is principal investigator of the Millennium Institute on Immunology and Immunotherapy in Santiago, Chile.
Thank you very much. And before all the acknowledgement, I'm going to connect my computer and I will. I know everything is working fine. So, first of all, I would like to thank the Universidad Austral de Chile for hosting us today. It's been a pleasure to be here, and also is really important for our Institute of Immunology and Immunotherapy start to foster and strengthen the collaboration, and also the activities that we do on immunology at the south of Chile, and also respect for all the regions in Chile. So also I would like to thank Dr. Calvo for her hospitality, of course, and for working with us with this like a challenge to do the first symposium in Valdivia, and we hope that this will be the first of many others. And also to all the speakers, and Dr. Juan Francisco Falconi, that is here with us from Pfizer, that is supporting this symposium, and of course Alex for making it today after all the problems with the connections, but it's fine. So I would like to work a little bit on a different project that we have been working. It's related to identifications of biomarkers of respiratory severity disease. We are coming from the identification of immunological markers, in order to try to categorize patients from their severity of disease early during infection, and that translated to a potential tool that could be used in most of the primary care services in order to help with the distribution sometimes of scarce resources that in countries like Chile sometimes are not available for everybody. So, first of all, we know that the respiratory tract infection are the main cause of morbidity and also mortality worldwide. Actually, 2.5 million of deaths each year are a consequence of our respiratory disease. And most of these respiratory diseases are caused by infection. And in countries like Chile, when we have a seasonality, usually what happens is that, of course, we have through the whole year cases of respiratory tract infections, but during the winter time, when we have colder months, usually the respiratory tract infection peaks, and with low or not very available resources of emergency rooms for all over the country, that every year we have the same situation. Sometimes it causes the stress of the primary and primary care services and hospitals. So most of the etiological agents of these respiratory tract infections are viruses. Not for all of them. We know we got vaccines to protect the population before the initial or the beginning of the winter. We know that right now we are starting the autumn in Chile, so everybody is encouraged to vaccinate against influenza, SARS-CoV-2, and in the case of babies, that the newborns, to be applied the nercimab. That is universal in Chile. But for several other respiratory viruses, we don't have any vaccine preventive or any other preventive measure to avoid or reduce the number of cases that can stress the health services in our country. But it's so important to keep developing and researching for getting a vaccine that can, for instance, for RSV and metapneumovirus, that can be applied for the most severe population, that is the newborns. Although we know that we already have the RSV for elderly vaccine and the monoclonal antibodies, we need for babies a more long-lasting mechanism of protections. And of course for bacteria also we have vaccines that also are usually concomitant to the respiratory viruses that cause also some important problems in the pediatric population, for instance, and also for elderly. But the use of antibiotics, we know that are in some cases questioned by the increased resistance to these mechanisms of protection. So what happened, as I mentioned before, is that usually, for instance, in 2016, and this is something that it hasn't been changed for this, at least 10 years, we have a lot of attention in one specific city in the center of Chile due to respiratory tract infections that leads to the waiting times from 3 to 6 hours before getting a medical evaluation. And usually the diagnosis of these respiratory tract infections are based on symptomatology only. And this is caused by, there are no methods for detection of respiratory tract infection, usually on the urgency or the primary care that is available for everybody. So molecular panels actually are not only in Chile but in several other countries that have health services as Chile are available only for a few number of people in the whole country. So this leads to the hospital collapse sometimes when there are very severe outbreaks of these diseases. So what we know is not only the presence of the infection. For instance, the diagnosis of respiratory viruses could identify or could be related necessarily for the severity of the patient at the time of the diagnosis. This is a representation of the alveoli, where there is a healthy alveoli in the lung. We have cells that have receptors for the detection of pathogens, and also a few immune cells. In most of the alveoli is free for the gas interchange. But after an infection, where in some patients leads to the infection of the most inner part of the lung, there is an activation of all these innate immune mechanisms that leads to the recruitment of cells in order to clear the infection. And this is essential to restrain the infections, but sometimes we have an increase in excessive recruitment of immune components in the lung that finally leads to the impairment of their principal function, that is the interchange of gases. And this has been associated with an increased production of inflammatory cytokines that are more balanced to the inflammatory side and less to the anti-inflammatory side. So it's not a well-balanced immune response that could lead a excessive recruitment of these cells and a poor clearance of these cells during the infections. This is an histology, a picture of a lung that is highly recruited and infiltrated by inflammatory cells, especially in young kids that can lead a more severe disease that require special attention, ventilation, and sometimes intubation in the worst cases. We expect that, and we would like, and usually it's the desirable situation, that the inflammatory response could be restricted at the time that the infection is in the lungs, and then detection of an excessive recruitment of these inflammatory cells, the production of anti-inflammatory cytokines, and then the clearance of these cells from the lungs, leading to healthy lungs and the recovery of the patients. So that is our goal in our research, is try to find a prognosis marker that could be increased in severe respiratory virus infections that can not only tell us that there is the presence of the virus in the, or the pathogen in the lungs or the airways, but also what kind of cytokines of inflammation is, or inflammatory response it's being produced during this disease. So what we have been working during this year is trying to develop several antibodies that can detect both viruses, bacteria that can lead to the production of the identification of proteins produced by those pathogens, but also antibodies against some inflammatory cytokines that can be detected in the nasopharyngeal swab. Why? Because this is the most accessible kind of sample that can be collected from infected patients, and actually from the emergency rooms there are the most simple way to get a sample that can lead us to the detection of infection but also inflammation. The good part of detecting antibodies or proteins with antibodies is that with antibodies you can generate any kind of diagnosis methodology, from the most sophisticated to the most simple, that are that can be applied depending on the setting of the clinical conditions and the infrastructure conditions. And we have developed several antibodies against the most common respiratory tract infections, also for bacteria, and we have identified in our research important and relevant cytokines that actually are elevated during the infection with respiratory viruses, as I'm going to show you in the next slides. So I'm going to talk a little bit about the respiratory syncytial virus. That is something that we have been focuses in this specific research. That everybody knows that this virus is the main cause of respiratory tract infection in children during the winter season. And the infection caused by this virus at early age correlates with respiratory hyperreactivity and asthma. So due to this characteristic, and also for the impact of the infection of this virus in this population, our aim was to identify in this specific population some inflammatory biomarkers of disease severity in the respiratory secretions that can be collected directly for the nasopharyngeal response. So we have, by the years, we have collected enough evidence from our side and also for all the people around the world working in this specific area, that when the respiratory syncytial virus infects the epithelial cells in the airways, produce several innate cytokines that are alarmins that can trigger inflammatory response. So it has been described that these kind of cytokines that usually are triggered in the epithelium by the respiratory syncytial virus could not lead to an effective response that can clear necessarily the virus, but induced excessive recruitment of inflammatory cells that can worsen the case. So in the case, for instance, of the recruitment of T cells due to cytokines as IL-6 can lead to the production of excessive or inappropriate levels in a specific time of IL-10 early during infection that can lead to the immune suppression of the respiratory tract, leading to bacterial co-infections and increasing the possibility to have pneumonia. In the case of IL-8, the recruitment of innate immune cells that can lead to and are associated with the bronchiolitis and disease severity, and also with ventilation requirements, hypoxia, and also asthma development. And some of the alarmin, that is the one that we have been studying during this research, is IL-33, that is associated with the recruitment of ILC2 cells that leads to the production of IL-13, the recruitment of eosinophils, and also the number of eosinophils of the recruitment of these cells that are not efficient for antiviral immunity can lead to the asthma development and mucus production, airway hyperreactivity, and inflammation, exacerbating the disease. TCLP also it's associated with the disease severity, and also IL-12 with T cells that leads to the skewing of a Th2 immune response that also exacerbate the possibility of developing asthma, especially when the infection occurs early in life, because it's been described that IL-33 in children's or in models that are early in life produce excessive production of IL-33 due to infections. So we did some clinical trials in Chile evaluating the correlation of some cytokines, for instance IL-3 and IL-12p40 in the respiratory secretion in some patients with bronchoalveolar lavage and nasopharyngeal aspirates, that correlates the number of or the level of these cytokines in the respiratory secretion with the episode of recurrent wheezing, a positive correlation. And also in the case of the fold increase of mRNA in the induction of or the presence of atopy in the respiratory secretion. Higher levels of IL-8 and IL-33 are found in the patients that are developing or have then been diagnosed with an AFOP phenotype in the respiratory secretion. So also there are later other disease that other studies, that have been correlating the presence of IL-3 in respiratory secretion with increased possibility to have a severe disease. And actually, after COVID-19, IL-33 was also described for several other, not only RSV and COVID-19 as well, but rhinovirus, influenza, adenovirus, methanovirus, a correlation with the most severe disease. So we also keep developing some clinical studies in Chile that were published in 2022 in by our groups, where we recruit patients from that were positive for RSV, and they have we collected nasopharyngeal swab and also PCR viral panel to identify the presence of RSV, and then to get the detection, actually, of the level of the cytokine in the nasopharyngeal swabs at the moment of the infection and the diagnosed, and then the follow-up of the patient to see the severity of the disease. So what we found is that after evaluating several cytokines, we found that some changes that were significant were again found in IL-3 and IL-33 and TCLP and IL-8. And especially in the case of IL-33, we found that the patients that were developing lower respiratory tract infection compared with the upper respiratory tract infections had higher levels in the respiratory secretion than the patients that were developing a mild disease. The same with IL-8. So we decided to keep working with this cytokine, and we evaluated the levels of these cytokines during winter in 2024 in several other clinical trials, both in Chile and Argentina and in Greece as well, with our teams here in the Hospital Sotero del Rio in Santiago, also in Buenos Aires with our collaborator Mauricio Caballero, and also in Greece with our collaborator Georgia Julia in the University of Thessaloniki, where we recruited hospitalized and ambulatory patients to evaluate with a higher number to validate the increase of this cytokine in the respiratory patients that were positive for several viruses, in this case not only RSV. And this is the overview of the study. We recruited ambulatory and hospitalized patients. All of them were selected to be 3 years and younger. And we used a multiplex technique as Luminex to detect the level of IL-33, that is a more sensitive technique, to detect the levels of IL-33 in RSV positive groups, a methanovirus positive group with other respiratory viruses and with co-infections. And what we observed is that the ambulatory patients, compared with the hospitalized patients, had consistently low levels of IL-33 compared with those that were hospitalized, both that were infected with RSV, with methanovirus, and with other seasonal respiratory viruses. So it's with this data, we demonstrate that, in fact, this cytokine has been elevated in the case of more severe disease, not only for RSV, but for other respiratory viruses, and actually some of them that are also have a higher impact in younger kids less than 1 year. And one interesting thing that we found is that also patients that had co-infections, either with RSV and methanovirus, and or RSV and rhinoviruses, we found increased level of or equivalent levels of IL-33 in respiratory secretion, both in ambulatory and hospitalized patients. So we think that this is a marker of more severe disease, with co-infections usually are associated clinically with more severe disease, especially in younger kids. So we are pursuing in keeping study what are the prognosis and the development of co-infections in patients that can be detected. But of course, it could be with all this data that we have gathered, both that we have antibodies against RSV, that we have developed in our lab, our research on IL-33, and also other cytokines that we, of course, further in the future would like to include, but we are working right now with IL-33, and we have developed as well our own antibodies against IL-33, and that in the case of anti-RSV antibodies, we have evaluated in clinical samples that work with a good sensitivity for the detection of RSV. How we can translate all this research in some useful tool. And that is what we have been doing. It's when we have these winter outbreaks, it's like our dream that we would like to achieve. Before the patients get to the medical evaluation, and this waiting time between 3, 6, or 12 hours in some of the worst days of the respiratory outbreak, we can provide a solution that can be an easy, fast, affordable technique that can allow us to rapidly take respiratory samples, know what is the virus, but also the level of the inflammatory cytokine in the respiratory tract. And we would like to provide at the clinician, when they have the opportunity to see the patient, already knowledge of the virus and the level of these cytokines in the respiratory tract infection, in order to decide what is the best management of this patient in the emergency room, either the discharge or the hospitalization or the observation. So using our tools, we have put together a multiple immunochromatographic script that we are intending and working that be less expensive and easy to perform, especially in rural areas. For instance, we are here in Valdivia, that is the capital of this region, but also there are several healthcare services primarily that are in rural areas that could benefit a lot with some tool like that. That can be rapid, in 30 minutes, give results, and not only the infection, but also the severity marker. So we have been working with a company in the UK that can produce and allow us to generate this device with the ISO regulations that can be used in clinic, where we can use the sample to mix with the antibodies that detect the RSV and IL-33, conjugated with colloidal bulk, and then migrate and give something like that. If it's positive for the cytokines, if it is positive for the virus, and also the control. And we have already patented this, at least presented a patent for this methodology, this idea. And what we have done currently, it's a device that can detect the two analytes, the virus and the cytokine. And we have been able to, of course, detect the recombinant protein, both the virus and the cytokine, and also in the case of cells that are usually in the laboratory used for the expansion of the respiratory syncytial virus, not infected cells, we see that the device is very specific no cross-reactivity. But in the case that we include infected cells, a lysate of the infected cells, we see a very strong response of the RSV production. Still, we need to see the IL-33, but I will show you data in the next slides. And also, we have evaluated whether how is performing this kind of test if we compare the level of CT or the thresholds of the qPCR with the number of plaque-forming units per ml. That, as we see here, lower the viral load is higher the thresholds cycle that detect by PCR, a molecular technique, the presence of the virus. And once we have increasing the level of the PFU, we see a decrease in the CT. And for low levels of the virus, we see that it's not detection with our technique, but from 10 to the 1000 PFU in a respiratory secretion, we have a higher level of the P protein, that is the base of the detection of this our device, and also we start to see the detection of this virus in the respiratory secretion. So we know already more or less what is the threshold level compared to the PCR of our immunochromatographic device. And also we have put together this test using in positive samples that we have recruited in our clinical trials, that for RSV positive patients that were diagnosed by PCR. As you can see, it's variable, some low CT, high CT, low detection, low CT, higher detection with our test, and also in cases when we see also positive for the virus and also for the IL-33 as well. So, as we detect or study the availability of diagnosis in, for instance, I would say in several countries like Chile, we know that in healthcare service, as I mentioned before, a very few percentage of the population has the possibility of have molecular panels that are really, really costly. And in public hospitals and primary care, usually we have a very few availability for molecular panels. Usually are immunofluorescence, that the sensitivity is okay, but still there is a time-consuming technique in some cases. And usually in the primary care and family centers, there are no laboratory methods available. So we would like to introduce our rapid test that could be cheaper, that our target is to have a sensitivity and a specificity equivalent to the immunofluorescence at least, and have been available for the primary care services that need the most. So in conclusion, the data that I showed today is that the inflammatory cytokines and viral proteins can be detected in the nasopharyngeal swab of children for simultaneous detection. We have validated that higher levels of IL-33 in nasal swabs are observed in children with lower respiratory tract infections associated with RSV infections. We observe also that IL-33 is increased in the respiratory secretion of ambulatory patients with co-infections, and we have developed new antibodies to detect these cytokines that could be used as a biomarkers associated with RSV diagnosis, and we have put together a prototype that could be used for this instance. This you can also, of course, revise with more detail our publications in this project, exciting project for me. And I would like to acknowledge all the scientific team, the past member of the team, all the clinical team in Chile, and of course our international collaborators, and the funding sources that have allowed us to develop this or try to advance this dream. Thank you so much for your attention.
Thank you, Dr. Bueno, for your impressive talk. Is there any question from the audience?
Turn on the lights. That's great.
Thank you, Susan, for the excellent and nice presentation. I was wondering about the I-33 because I understand, and correct me if I'm wrong, that they have kind of dual roles, promoting Th1 and Th2 responses depending on the cytokine milieu. So in this case, especially when you have co-infections, do you think that will lead to an exacerbated Th1 response that is not productive, like what happened with the cytokine storm in COVID? Or to be the other way, like having an exacerbated Th2 responses that will lead to all of the other problems without having a productive immune response against the infection?
Okay, thank you very much for the question. Yes, the data in all the research, both in humans and also in animal models, have shown that the Th2 skew of the immune response is in most of the cases the problem with the exacerbated inflammation, the mucus production, and also the correlation with asthma in the future. And it seems like the RSV and also the metapneumovirus is causing a specific signaling in the epithelial cells leading to increased production of alarmins that are more associated with allergic disease and the recruitment of basophils and eosinophils. So, and ILC2, that is the most recent research that have been done in this area.
Any more questions?
Thanks for the presentation. A quick question, because you have been working with different respiratory viruses and
That's
bacteria. So I would like to know if you have seen an increase of an association between also IL-33 and other respiratory viruses. I was thinking in what happens if I take the test and I see an increase, for example, in IL-33 and not the signal of the virus, for example.
Yeah.
And my second question is, because you have other antibodies, if you are thinking to do a test to detect other viral or bacteria that cause very huge problems during winter, right, in respiratory systems.
Thank you, Maria Ines, for the question. Yes, both are very excellent questions, and also we are working on that. In the case of IL-33, indeed, we have seen an increase in viral infections. Usually, if we go for a bacterial infection, especially bacterial infection, we don't see this increase. But for the damage, it seems like is caused by viral infections, is an increase in the IL-33. And the interesting thing is that, as you mentioned, we have seen cases with are negative for RSV or metapneumovirus that have increased level of IL-33. And we would like to validate that actually could be a biomarker not only of severity but also for viral respiratory infection. And indeed, we have been trying to put together, was very ambitious, our first approach to put all together in a single test, at least 5 viruses and 2 cytokines, but it's absolutely very, very difficult. So, but the idea is actually to develop a customized test. For instance, in the case of adults, it's more important in, actually there are right now, actually by Pfizer, actually, the co-detection of SARS-CoV-2 and influenza that are more prevalent in this population, but in kids it's more important to detect early the respiratory syncytial virus and the metapneumovirus. That is what we are trying to do right now, and also the cytokine.
Okay, thank you very much for your participation today, for your talk. Now we have a lunch break, so we'll meet again in this room at 2:30. Thank you.
The next speaker is Dr. Pablo González. He is full professor of Facultad de Ciencias Biológicas, Pontificia Universidad Católica de Chile, principal investigator of the Millennium Institute on Immunology and Immunotherapy in Santiago, Chile. The talk is restoring dendritic cell function to enhance antiviral immunity against herpes simplex virus.
Thank you very much, Mr. Calvo. So it's my pleasure to have the opportunity to tell you a bit more of the research we're doing at my lab. Just rapidly change the input, so maybe that's what's causing the noise. Seems to have worked. Okay then, so you should be able to see the screen online and also receive it right here. Quisiera saludar primero a la audiencia y agradecerle la bienvenida a un almuerzo, pero no hay almorzado muy pesado y les de sueño, la alcalina, así que vamos a ir lo más dinámico posible. Ahí esto lo quiero mostrar, así que voy a empezar y por supuesto el idioma inglés por el idioma oficial del simposio. So I'll switch back to English. This is our official language of this symposium. And as I mentioned before, I'm very glad to be able to show to you some of the work we've been doing in my lab regarding this specific aspect, which is restoring the dendritic cell function to enhance antiviral immunity against herpes simplex virus. So let me introduce rapidly herpes simplex. There are actually two viruses, herpes simplex type 1 and type 2. They are viruses that infect for life, so once you get herpes simplex, you never get rid of it again. So it's forever. It's not as simple as the name suggests. So simplex, because it's one of the first herpes described. It's actually a complex virus because it has more than 80 genes. So it has many mechanisms for evading the immune system. And that's why also it can persist in our organisms for life. So something that's very important and people tend to forget is that more than 70% of the population is positive for HSV. And within these 70%, something that's always been very impressive for a matter of study is that only 10% of those that are infected actually have permanent recurrences, permanent symptoms of HSV. And of course, part of the work that we are focused on is helping those that have these recurrent lesions, which is very uncomfortable and certainly it impacts quality of life. So something very interesting also within those that have manifestations is that 24% of those infected have recurrent skin infections. So they will have every month or once a year, between 1 to 12 recurrences a year. And even though there are antivirals for herpes simplex, mainly acyclovir, the effect that it has in reducing the length of the lesion is very small. So actually, a normal lesion will last 7 days in the skin. If you apply acyclovir every 4 hours to the lesion, you only reduce it from 7 days to 6 days. So you still have 6 days of lesion, which is not very significant at the biological level. It is significant mathematically, but at the biological level for the person who is affected, is not very biologically significant. So we need better solutions, we need better therapies or approaches for those that have these recurrent episodes so that we can help them have a better quality of life. So what makes a good antiviral immune response? Well, the dogma says that an optimal immune response is based mainly or mostly or frequently for viruses based on neutralizing antibodies. And these antibodies, they're produced with the correct isotype an amount when they are correctly trained by CD4 T cells. CD4 T cells that have the appropriate phenotype, hopefully a T follicular phenotype or Th1 phenotype. And those T cells, they get that phenotype because they're properly trained by the dendritic cells. So if you go back to what is at the basis of a good antibody response or a good T cell response, the basis of that response is at the dendritic cell level. And that's why the dendritic cells have won a Nobel Prize, because they're very relevant as the yin-yang in inducing, promoting, or regulating the immune response. This is, as you say in Spanish, la madre del cordero, right? Here in the south. So of course you have also, if you want CD8, the cytotoxic T cells, you also need these dendritic cells. So dendritic cells are key for this optimal immune response. So of course what happens in the context of HSV? Is HSV friendly with dendritic cells? Are dendritic cells able to present antigens to T cells once they interact? Well, the outcome reported in the literature is actually quite bad. So the virus will actually modulate different functions of dendritic cells to avoid antigen presentation. So you can see there, it will promote, the virus will promote the dendritic cell to produce, will promote the production of pro-inflammatory cytokines, which are not useful for disease resolution. It will inhibit autophagosome, which is needed by the dendritic cells to process viral antigens. It will inhibit MHC class 1 presentation, which is needed to activate cytotoxic T cells. It will inhibit the expression of co-stimulatory molecules on the surface of the dendritic cells, which are also needed for T cell activation. And if that is not enough for you, the virus will also kill the dendritic cell. So it's like a cat playing with a mouse, so it'll mess around with the functions of the dendritic cell, and on top of that, it will kill the dendritic cell. And again, if we go back to the dogma, if you have no dendritic cells, you'll have poor T cells. If you have poor T cells, CD4 T cells, you will have poor B cells, and you'll have poor cytotoxic T cells. So the focus of my laboratory is mainly on restoring dendritic cell function. What can we do to make these dendritic cells manage HSV infection, hopefully process HSV antigens, and induce an optimal T cell response? Which is the optimal T cell response? We don't know yet, but what we want first is restore dendritic cell function so that we can think on the best T cell response or T cell or immune response against the virus. So we've been doing this through different approaches. I want to bring you to the basis of what we've been doing the last, I'd say, 9 years. So while we were studying a mutant virus, which is here called Delta GD, on why was it such a good immunogen or vaccine for inducing protective immunity in mice, we came up with this piece of data that for many years we did not pay much attention. So we were mainly focused on looking what was so good of Delta GD that made it a good vaccine, which is attenuated virus. So we're looking at different aspects. We looked at many aspects. We actually never found what was it that made it such a good vaccine. And we had this piece of data that actually we were much interested because we were looking at the Delta GD virus, which shows that the wild-type virus, it actually induces high levels of this spliced XBB1 messenger RNA, which belongs to the unfolded protein response axis in dendritic cells. So there's this cellular response called the UPR that helps cells manage stress of high translation levels. And there are actually 3 pathways, and there's one specific called the RE1 alpha that's you can read out through this measurement here that tells us that this pathway specifically is super activated in dendritic cells infected with the wild-type virus. So we found literature actually that was quite interesting that would suggest that viral infections can trigger the UPR response through IR1 alpha to induce cell death. So we went in and just inhibited the IR1 alpha axis through 2 inhibitors, 4-MUHC and MKC. And long story short, what you can see here is that if you look at something so basic as cell viability, dendritic cell viability infected with viruses, so this is non-infected dendritic cells. These are dendritic cells infected with HSV1, infected with HSV2. You can see that the virus kills the dendritic cells. You have 20% of the culture at 24 hours. If you add form HC or MPC, you recover viability. You can see this here, viability. The opposite is also true. If you see increased caspase activity upon infection with no treatment, now when you apply the treatments, you see reduced caspase activity. So you have increased viability, reduced apoptosis. And what does this do to the dendritic cells? Well, many things. If you look at the function of the dendritic cells, you can see that these dendritic cells now migrate much better. So if you inject them in the foot pad of the animal, and you go to and look at the lymph node that's draining that zone in the foot pad, you can see that upon normal infection, very little dendritic cells make it to the lymph node because they die in the foot pad. They're infected. But if you treat with these drugs that inhibit iron alpha, those dendritic cells are alive and they can migrate to the lymph node. And you know that in the lymph nodes you have the T cells that can interact with the dendritic cells, be activated, and induce adaptive immune response. And if you look also in vivo, you can see that actually this is true. CD8 T cells and CD4 T cells are activated in vivo when you treat the dendritic cells with these drugs. And these are T cells that are specific for the virus because they're transgenic with TCRs for HSV antigens in MHC class 1 and MHC class 2. Okay, so what happened after that? We found literature that would say that actually there was a connection between the UPR response and lipid droplets. So actually there's a paper on dendritic cells in the context of cancer that says that a cancer environment or tumor environment can induce lipid droplets in dendritic cells and impair their function. It makes them like foamy dendritic cells, like foamy macrophages, foamy dendritic cells. So we ask ourselves, is this happening as well in HSV-infected dendritic cells? So we did RNA-seq and also profiling of lipid metabolism pathways and found that these dendritic cells that are infected with the virus have significant differences in the expression of genes related to lipid metabolism. So there's something happening with lipid metabolism in these cells. And actually you can see some pathways, adipogenesis, glycerolipid and glycerophospholipid synthesis, they're altered and actually increased. And you go look at confocal electronic microscopy, you can see that the infected dendritic cells actually have these kind of vacuoles. For many years we thought they were vacuoles, but they're actually lipid droplets. They're vacuoles full of lipids. And even though electron microscopy cannot demonstrate this fully, if you look by confocal microscopy with a probe that stains neutral lipids, you can see that the infected dendritic cells have accumulated lipid droplets or structures that are rich in neutral lipids. Okay, you can see there's an increased amount, increase in size, and these dendritic cells have this issue. What's interesting is, again, if you go long story short, there's many assays we did in this study also with the T cells, but long story short, if you now go in vivo to see if you can improve the functions of these dendritic cells by altering lipid metabolism, you can see here that treating with avastinib, which is a specific inhibitor of one of the enzymes involved in lipid metabolism, you can recover the migration of dendritic cells to the lymph node. And you actually look at the different types of dendritic cells, you can see this is mainly important for dermal dendritic cells, which are better at activating CD8 T cells over CD4 T cells. If you look at T cells, whether they're activated or not, you can actually see that this capacity with this drug of these dendritic cells treated, they're now able to activate CD8 T cells in vivo. Not CD4 T cells, surprisingly, but they can activate CD8 T cells in vivo. Okay, what else? So then we found additional literature that would suggest that you can actually inhibit UPR-mediated death by treating with or promoting the expression of an enzyme called hem-oxygenase 1. It's an enzyme that you can induce with different drugs in practically any cell in the body. You can actually, this is what the enzyme does. So the enzyme, if it finds free heme, which happens in damaged proteins, this damaged heme can relate to production of oxygen radicals that can damage the cell. So these species, such as reactive oxygen species, oxygen peroxide radicals, can induce the expression of this enzyme that will process the free heme and convert it into carbon monoxide, biliverdin, which is converted to bilirubin, and free iron. And surprisingly, For this, I would need several slides to explain, but surprisingly, these products, they're actually cytoprotective. So the amounts, at the levels of amounts of carbon monoxide this enzyme produces, those levels are actually pro-life instead of toxic. They promote actually mitochondrial function and can promote cell survival. So we can induce this enzyme with two drugs or different drugs. Here with COPP, cobalt protoporphyrin, we can induce hemoxygenase expression through its transcription factor NRF2, or we can inhibit the enzyme with thin protoporphyrin directly by inhibiting the catalytic activity of the enzyme. So again, let's go back to dendritic cells infected with the virus. We can treat it with either COPP, pro-HO1, or with SNPP, an inhibitor of HO1, and look what happens with these cells. Let's go again to the basics. Let's look at, for start, viability. Again, uninfected dendritic cells, they look healthy. Whether you treat them or not with COPP or SNPP, they're alive, they're happy. You infect them with HSV1 or HSV2, they die. High percentage. You treat with COPP, you recover cell viability. You treat them with SNPP, nothing really happens. So COPP is actually promoting the viability of these dendritic cells. So actually there might be a connection between HO1 and the EPR response because we're getting increased viability with these drugs. The same here applies for caspase-3. It's the opposite. If no treatment is added, you get increased caspase activity, which means increased apoptosis. If you add COPP, you reduce apoptosis, you reduce caspase-3 activity. Okay, we have a live dendritic cell. Does this mean anything for T cells? It does, because you can induce, for T cells with different phenotypes, you can use Tregs, you can use Th17 T cells. You can actually induce the double positive Treg Th17 T cells. And actually, if you measure IL-17 in the supernatant, you can detect the supernatant IL-17. So these dendritic cells, they're alive, they can activate, for T cells, and actually they can give them a phenotype of Treg Th17 T cells. Is this good or bad? Let's see. So first of all, can we detect these T cells in the skin upon infection, upon skin infection? So actually you can find these T cells, they actually have a similar phenotype. So this is 10 days post-infection in the skin. You can see that these T cells, T cells, they're activated in the skin. You find activated virus-specific T cells in the skin. And these T cells, they don't have a strong Th2 phenotype, but they do have mainly a strong Th17 phenotype, a strong Treg phenotype. That means that the phenotype we're observing in vitro on Treg Th17 is also observed in vivo in a skin lesion in the mouse model. And of course, what matters the most, what happens if with the score, with the disease severity of these animals when you treat with COPP, what we observe, you see, look here at the curve, this is clinical score in time. You can see that HSV alone, you get high scores, which last for several days. If you add COPP in red, you get reduced score, and it also shortens the duration of the disease. So as a means to integrate both the disease score and the duration of the disease, you can do the area under the curve. You can take all the area under the curve, which integrates disease severity and length. You can graph that, and you can see that COPP reduces overall the severity and the length of the disease. Okay? And if you look at a piece of skin, and you measure the amount of virus in that piece of skin, you can see that those treated with COPP, they have less virus in that piece of skin. So you get less viral replication, less disease score. Okay, and last thing I want to show to you is something more recent. We've been working on the lab. It's another connection. We found, again, interesting literature that would relate UPR-mediated death with the induction of cyclooxygenase in immune cells. Okay, so there's a relationship between IR1 alpha in prostaglandin production. So the story behind, or the details behind prostaglandin production in our cells is quite complex. This is, again, a long story short. So you have phospholipases can take phospholipids from the membrane. They transform this into arachidonic acid, and then COX-1 or COX-2 will process this arachidonic acid into prostaglandin H2, which then is processed into prostaglandin E2, which is the one we most know because it's related to pain, to the intake of anti-inflammatories or anti-pain drugs, acetaminophen, aspirin, celecoxib, etc. And the product, prostaglandin E2, actually works through its receptors called EP1, EP2, EP3, EP4. And each of them has quite complex signaling pathways through different transcription factors, which overall modulate inflammation and many other processes. So, what happens in the dendritic cells infected with the virus? We looked for first by qPCR what happens with the levels of expression of COX-1 and COX-2, which are the genes PTGS1, PTGS2. So COX-1, no significant changes besides a significant reduction in the condition of infection with HSV1, but this is quite surprising for us because we found that upon infection of the dendritic cells with HSV1, we get this huge increase in the expression of RNA messengers for COX-2. So the virus is inducing COX-2 expression in the dendritic cells. If you look at the cells by confocal microscopy, stained for COX-2 in red, you can see that those cells infected do have increased levels, at least qualitative in this image, of COX-2 compared to control cells. If you look at the levels of proteins by Western blot, you can see that cells infected with HSV will have an increased level of COX-2 in time. So you look here at 18 hours, you can see that cells infected with the virus have increased levels of COX-2, same here compared to mock-infected cells. So the virus is inducing increased levels of COX-2, at the level of RNA messenger, at the level of protein. What about the activity? Increased levels of protein doesn't mean increased activity. So we went in and measured the activity of COX-2 through a probe that allows you to measure how active is the enzyme in the cells. So you can see here that vehicle-treated dendritic cells infected with the virus have increased activity of COX-2. Actually, we know it's COX-2 because if you treat with celecoxib, you see that the levels drop dramatically. And actually, also having high activity of COX-2 doesn't necessarily mean high levels of prostaglandin E, because there's several pathways or enzymes in between. So we measured prostaglandin E2. You can see again that in the case of infection, the cells have increased levels of PGE2, and when you treat with celecoxib, they fall dramatically. So most of the PGE2 comes from COX-2 induced by the virus in these cells. And again, the same question, what does this mean in terms of function for dendritic cells? Do these high levels of COX-2 mean any impact on cell viability, on virus production by these cells, or on their capacity to activate T cells? And the answer is as follows. If you measure amounts of virus produced in these cells, they actually don't change. So regardless if you treat or not with celecoxib, you have the same levels of virus produced. This actually may be good because you want optimally viral antigens to be expressed in these cells so that the dendritic cells can present them to other cells. So there's no changes there. And what happens with the viability of dendritic cells? You can see there's an increase in the viability. So these are again non-infected cells, cells infected, there's a reduction in viability. You treat with celecoxib, you recover viability. So these dendritic cells now are better alive than they were before. So actually, this also shows this is for celecoxib. This is an inhibitor of COX-2. If you apply an inhibitor of COX-1, you can see nothing happens. So this is specific of COX-2. It's COX-2 that's mediating the detrimental effect on the cell. Is it the prostaglandin E2 from COX-2 that's inducing this cell death? There's only one way to know. So we measure cell viability and add different amounts of PGE2, and you can see that the viability of dendritic cells decreases as you increase the amounts of PGE2. So these cells are sensitive to cell death due to prostaglandin E2. And is it the prostaglandin E2 coming from COX-2? Yes, because if you add celecoxib, you can recover this viability even in the presence of PGE2. So this PGE2 plus celecoxib. And is it mediated by the receptor of PGE2 through the EP receptors? It is, because if you treat with inhibitor of EP2, EP4, or the combination, EP2 plus EP4, you get a recovery and in dendritic cell viability compared to no treatment at all. So we have again a drug that inhibits a specific enzyme within these cells that can recover the viability of these cells. And what is of interest again for us is whether this increase in viability will help or not the function of this dendritic cell. So again, we go and look at migration of dendritic cells. Dendritic cells are treated with virus, with or without drugs. Then they're injected in the footpad. And then we go and look in the lymph nodes whether these dendritic cells are migrating or not. You can see if no treatments applied to cells infected, there's just basal migration, just as non-treated cells. But if you apply celecoxib, you now have these dendritic cells that are infected, they're actually migrating to the lymph node. So they can now probably see T cells to present what they captured. Do these cells change in phenotype? So we did here an array of 110 inflammatory molecules that you can measure with the supernatant of infected cells. So this shows you here those inflammatory molecules that are upregulated in red and downregulated in blue between non-infected and infected dendritic cells. So you can see the infection does induce changes in inflammatory molecules. And if you add to the infected dendritic cells celecoxib, you have a complete change in the phenotype of the secretory phenotype of those inflammatory molecules that are secreted to the environment. And that would most likely impact the capacity of the dendritic cell to infiltrate different types of immune cells. So we actually measured that. This is an assay where, again, you treat dendritic cells in vitro, with or without treatment, you inject them in the footpad, but now you recover the footpad. You take the footpad, and you look by flow cytometry what cells are infiltrating the footpad that has these dendritic cells. And you can see, in orange, these are the immune cells that are upregulated by viral infection, and then in blue, those that are modulated differentially when they're infected and treated with celecoxib. So celecoxib will induce a change, significant changes in some cell types that migrate towards the site of infection because they have a different phenotype now. And hopefully, our expectation is that that phenotype is pro-activation of T cells that serve antiviral immunity. So the last slide, or before last slide I'm going to show you, is actually activation of T cells. This is in vitro dendritic cell T cell co-cultures. So you look at virus alone, there it is, in orange, it's very low activation. Now you treat with celecoxib. In blue, light blue, you have an increase in CD8 T activation. You can see this at the level of IL-2, the level of interferon, also differences at the level of surface markers that are indicative of T cell activation. You can also see this in vivo. Again, looking at activated T cells, you can see this significant increase in activation of T cells upon treatment with celecoxib. This is in terms of activation markers and in terms of proliferation. And the last slide, regarding what matters most, what happens with the lesion. So again, this is an in vivo model of herpetic simplex lesion. So no treatment, just infection, high score for a long time. If you apply celecoxib, lower score, shorter time. If you take the area under the curve, you have here celecoxib gives you a better output than infection alone. So we are some way ameliorating the immune response or the response to the virus. And if you measure amount of virus in the skin, it is as effective as treating with acyclovir, which we're not doing with an antiviral. So as a conclusion, as I showed in the first slides, HSV activates a UPR response, specifically the IR1 alpha axis or pathway. If you inhibit that pathway, you get recovery of dendritic cell function, viability, T cell activation. It seems that that activation of that pathway is related also to lipid droplet accumulation in dendritic cells, which makes them non-functional. So also attacking those metabolic pathways of lipids, you can also recover some activity. You can also attack this pathway through the induction, pharmacological induction of HO1, which serves also as a mechanism of recovery of the dendritic cell viability and function. And lastly, but not least, there's a role for COX-2 in the function of these dendritic cells that can also be modulated with commonly used drugs such as celecoxib or the COX-2 inhibitors. And their overall message to take away home is that we should try to test such types of drugs directly on the skin as a method for inducing increased immunity against the virus. So think about it, when you have a reactivation of the virus, you have viral antigen, you have the immune cells. So the only thing you need now is conditions that favor that those immune cells, in the presence of that viral antigen, are capable of processing and presenting adequately those antigens. And that can be given by a drug that now gives the dendritic cells license to support or control viral-induced death and promote T-cell activation. So with that said, just thank my laboratory team, many young students, talented students working on their projects on different stages of their training, undergrad and postgraduate. Here are the main students involved in the data I just showed, many other members. Also many thanks to collaborators, those collaborators with whom we interact many times a week, times a day, constantly. And also collaborators from other institutions. Of course, always thank the funding sources. So thank you very much. I'll take any question you may have.
Thank you so much for your talk. I was wondering if you look a little deeper in the case of the HO1 induction, where you saw that the T-Rex and the Th17 went up with the treatment, because those two populations supposed to be like antagonists. So what do you say about that, or how do you explain that those both populations are increasing in that condition?
So actually, that was reviewer 2 who asked for those experiments. Was it you? So
No, it wasn't me.
okay. So we were asked actually. We were surprised with seeing the Th17 phenotype, which usually is very limited to Klebsiella infection or very pathogenic scenarios, multiple sclerosis. It's not a phenotype that you usually expect or desire or would be looking at, particularly in the context of viral infections. But we saw it, and we also saw the T-Rex, which usually is not that frequently looked at, but we saw it also increase. And then this reviewer suggested, based on literature, it was very well documented, that there's a link between T-Rex and Th17 T cells. They can revert from one phenotype to the other, and that's why we actually looked at the double positive. And actually, we were surprised that a big proportion of those T cells were actually the double positive. So it might be that they start as Th17 and then transition to Treg. That's something we have to look into closely, and also make sure that actually that phenotype, the Th17, is seemingly, and according to what we saw, is good for resolution of the lesion. So we have to confirm that in other contexts and explore that more in deep.
Okay. My question was going on that matter. How did you see it? Was in with flow cytometry, and if you saw that they were double positive, because that can make some sense.
Yes.
On that.
Yeah. So
and
we looked 2 time points, 4 and 10 days, and so actually it first comes in as inflammatory and then transitions to more Tregs.
Yeah.
So it seems that there is a dynamic also of control of the lesion, resolve virus, and then resolve inflammation.
Yeah.
So it might be actually a good thing.
Thank you.
Look into that. Yes.
Very nice. In the mouse study, how were you giving Chile2026? Was this systemically?
Which case? In the case of the clinical score in disease?
In general, I guess what I'm trying to say, is there any even anecdotal data in humans that people that take Chile2026 for?
We haven't seen. We actually did a topic. So we formulated with the pharmacy. They formulated for us celecoxib in cream. And the only other study where we found that celecoxib has been formulated in cream is for arteriosclerosis, for rheumatoid arthritis. Yes, the clinical trials are running. They are using celecoxib cream, but it's not sold. You can't find it. It's not something that you find in the pharmacies. Nobody is selling it for any purpose. So we think that the topical application is the most optimal. We haven't tried the systemic. We should. It's what's available, right? And we don't know whether it's as effective as topical celecoxib.
Right. And related to that, you showed all this very nice data with HSV1. Is it known whether other herpes also have a similar bad effect on BC, or?
Yes, so HSV1, HSV2, for a long time in some of our studies that I did show have HSV1, HSV2, because we've been very interested in the lab in comparing both viruses. For many years there was literature mainly on HSV1 or 2, and they were generalized to both viruses. So we started taking practice of always assessing both viruses in our assays, as here. So it's double the work, it's double the expenses. So more lately, we've been focusing more on HSV1, but I would love to look at the in vivo effects and the drug effects in the context of HSV2 and the genital infection model, which we have established in my lab. It's just a matter of time and cost. We have simplified the studies to more common clinical manifestation, which is the skin manifestation over the genital infection model. Now we should.
Thank you, Dr. Gonzalez.
My pleasure.
The next talk will be presented by a local speaker, Dr. Dianara Bustamante. He's an assistant professor of the Facultad de Medicina, Instituto de Microbiología Clínica, Universidad Austral de Chile. The talk is From Traditional Medicine to Cost Antiviral Defense, Plant-Derived Molecules Targeting the NRF2 over HO1 axis to restrict herpes simplex virus infections.
Ya, muchas gracias por la invitación. Hoy les voy a presentar algo de nuestro trabajo que venimos haciendo recientemente en los últimos años. So, thank you very much for the invitation. Today I will show you our most recent research in the field host and virus interaction, and our work about the plant-derived molecules that have antiviral potential against herpes simplex virus infections. Our model in the lab is a strain of HSV1. The herpes simplex virus type 1 belongs to the Orthoherpespiridae family and is one of the 9 herpes viruses that infect humans. And this virus is transmitted between people by direct contact with infected skin or mucous membranes infected. So it's very easy to acquire this infection. The virus has affinity, a high affinity, with keratinocytes and sensitive neurons. So this virus established a latent and persistent infection during the life of the host. Between the main clinical manifestations, we found the herpes labialis, also complications, clinical complications, for example, encephalitis or conjunctivitis related to blindness caused by pathogens. It's one of the virus most spread in the population. 67% of the people in the world is infected by the virus. Produce genital lesions, and it can be transmitted during pregnancy or during the birth. And it's involved in common cause. It is a common cause of acute encephalitis in adult people, even in immunocompetent persons. So when the virus infects the host, it overcomes a series of barriers set up by the host to limit the spread of the infections. So the most earliest response to the host cells against the virus correspond to intrinsic ones. This barrier, by some authors, can be considered interchangeable term with the concept of innate immunity. Today, we distinguish the response intrinsic respect to the innate response. Because this kind of response corresponds to cytoprotective and cell-autonomous programs deployed for all cell types in the body, maintaining two main functions: protect the cells and restrict the spread of the virus, interfering the viral replication very quickly when the virus penetrates cells. So some of them response involve response that require the function of the endoplasmic reticulum, like UPR or IRAD response. Also, autophagy and apoptosis is included in this group of response. cellular components in double membrane autophagosomes. So these autophagosomes are fusioned with the lysosome to break down the cargos. Curiously, many viruses are able to hijack this cellular response, including herpes virus. During my postdoctoral training, we study the role of autophagy or xenophagy for viruses or pathogenic cargos. We study the role of autophagy in the management of the HSV1 infection in epithelial models. And we found in epithelial cells that the autophagy is activated to restrict viral replication, mediated by receptor or autophagic receptors. These proteins act as shuttles for the cargos, leading to the cargos to autophagosomes. Also, we found that the virus has a viral factor named US11. This tegoman protein is able to slow down this cellular process, slow down the correct degradation of the receptor, autophagic receptor. So this virus is able to hijack autophagy in epithelial cells. Respect to the antiviral drugs, since 80s, there are many compounds of drugs able to restrict the DNA replication in a specific form, interfering the DNA replication of the virus. However, the bad news is that the patients under long-term treatment with these treatments increase in the last years are increased the drug resistance due to mutations in specific genes in the virus. So the problem is increasing in the world. It's a public health concern, and since 20 years, have been studied many compounds derived from natural source with antiviral activity against herpes viruses. So these findings support the idea that in the herbal medicines we can find source for future novel antivirals. So during my postdoctoral training with my lab, we start a new line of research in native Chilean plant known as Soralea glandulosa o Mapudungun culén, o hierba de San Agustín. This plant is a bush that grows in 6 regions of Chile, mainly in the Central Valley. And in the popular medicine, and the traditional medicine, is recognized for the properties, antipyretic properties, antibacterial properties, and wound healing properties. And is used in the traditional medicine as poultice or infusions. Also, is used as an ingredient in an alcoholic drink known as ponche de culén. It's a drink very aromatic drink in Christmas time in the Central Valley. What science tells us about Julen? More than 10 years ago, my collaborator Alejandro Madrid published this paper where Alejandro found an antioxidant property in the exudate of the plant. When studying the composition of the exudate, he found bakuchiol bakuchiol as an abundant aromatic compound in the exudate of the plant. Bakuchiol is an isoprenoid phenolic, phenolic isoprenoid. 2 years later was found an antiviral property in the other side of the world, and this is an Asiatic paper. The investigators found an anti-influenza activity. The authors involved the activation of NRF2 pathway, mediating this activity, antiviral activity. NRF2 is a signaling pathway related to alleviate the oxidative stress. So in basal conditions, NRF2 is hijacked and ubiquitinated by KIP1. So this protein has a short half-life in the cell. So when the cell is stressed, under oxidative stress, NRF2 is released in the cytosol and can translocate to the nucleus to perform the activity as transcription factor and stimulate the expression of antioxidant proteins as oxygenase 1 and others. Interestingly, NRF2 is impaired during viral infections. Many viruses can impair this signaling pathway, and on the contrary, is an antiviral therapeutic target that must be validated because in the case of some respiratory virus, when by different approaches the signaling is activated, so the authors have found potential antiviral, including the paper of anti-influenza. So we have as model, in vitro model, keratinocytes, because the keratinocytes are the main cell type in the epidermis. In the epidermis is mainly for the virus, and is skin, and skin is the main barrier from environment threats. So in hacked cells, human keratinocytes, we test bakuchiol in different concentrations, and we found an increase in the levels of NRF2. Also, we tested the exudate and extract in dichloromethane of the flowers of the plant, and we found an increase in the levels of NRF2, replicating the effect of bakuchiol, the molecule, the isolated molecule. Also, we test as positive control of the assay sulforaphane, a drug very accepted as NRF2 inductor. Sulforaphane is obtained from cruciferous plants. So sulforaphane increase the levels of NRF2, and when we study the nuclear translocation, sulforaphane and bakuchiol are able to induce the translocation of NRF2. Later, we study the activation of the transcriptional activity. First, we prove the activation of the transcription factor and the target genes of the root. So, at levels of proteins, we found a specific and high potent effects on MOC-genase 1. It's interesting because the extract result very potent inducer of the roots, also bakuchiol. Also, we test the stability of the protein in the time, and cyclohexymal time course, we found an increase in the stability of the protein in the cell. So, when the protein remains more time in the cell, has more time to act as transcription factor. And we compare the remaining with MG132. This is a proteasome inhibitor, and we found an increase in the stability. Finally, in this story, we are going to see the possible interaction with KIM1, and the predictions in silico by molecular docking suggest that BAC inhibits the protein-protein interaction between KIM1, the hijack protein, and NRF2. So, seems to be BAC binds to KIM1 in the domain where NRF2 is ubiquitous. So, with this data, we have this model, and we think that Quillen is able to activate this NRF2 signaling pathway, and as consequence, MOC-genase 1 is a really good target of the potential of the plant. So, we test the effect of this plant in the progression of the infection by HSV1. Interestingly, the virus during the progression of the infection in the period of 24 hours in Hackett cells, we found a downregulation of the protein, even in the translocation in the nucleus. So maybe this virus also impairs this pathway. Interestingly, bakuchiol, when we treat the cells with different approach in different stages of the infection, we find that when we pre-treat the cells with bakuchiol, and after we infect and keeping the drug in the system, bakuchiol is able to reduce the viral titer in all stages, and is seen to be better when we apply the drug as pre-treatment. After we test mixing the drug with acyclovir, the standard antiviral drug, and we found that bakuchiol seems to be improve the antiviral effect of acyclovir in vitro. Even bakuchiol itself is able to reduce a lot the viral titer of the virus. Finally, in the last years, we work in HSV2 infection. It's interesting because it's related to genital infections, and the prevalence is higher in women than in men. In this model, using the strain G of the virus in the model of keratinocytes, we compare the kinetics of infection, we characterize the kinetics by qPCR and by viral titers, and also using immunoblot to prove the markers of the HSV2. And we found HSV2 in keratinocytes is more productive than HSV1. And we test also bakuchiol, the extract of culen and other compounds derived from culen, and we found a nice result because the extract in HSV2 reduced the viral titer in a similar way to acyclovir. So in this case, the pre-treatment of the extract reduced the viral progeny of HSV2 with a similar efficacy as the antiviral acyclovir. So, as final conclusions, both extract and bakuchiol induce activation of the NRF2 hemoxygenase 1 signaling pathway in human keratinocytes. But bakuchiol positively regulates this signaling pathway in two levels. At level transcriptional, increasing the activity of NRF2, and at level post-translational, according to our prediction, also block the protein-protein interaction. So, seems to be culen is a native source of molecules able to limit the viral replication in two species that infect humans. So we demonstrate the efficacy in vitro of these molecules, reducing the viral infectivity in human tissue cultures in a model that is natural host of these viral agents. So we thanks to my students and Paula Salazar, my colleague Paula, for the technical assistance, my collaborators in Chile, in Argentina, Antonella Locino, y the source, funding source in Chile. So we are very happy because this year we start a new project, Fondesit de Iniciación, to study to follow these studies, exploring this signaling pathway as a possible host intrinsic response. So many thanks for your attention.
Thank you, Dr. Bustamante. Any question?
Thank you very much for your talk.
I was wondering, there are some data related to the role of H1
in probing carbon monoxide production in the clearance of viral infection, especially herpes simplex. Dr. Gonzalez has a bunch of data. We are also generated with the production of carbon monoxide derived from bacteria, and also your data. So, what do you could be the mechanisms that do you think are related to the impairment probably of the replication of the clearance of the viral?
Yes, it's a very interesting question because until now we found an increase in the expression and in a level protein, but it's necessary to explore the explanation, the link with reducing the viral progeny. Hasta ahora no lo hemos podido explorar. Bueno, recién voy a tener financiamiento para seguir avanzando, pero de acuerdo a la literatura más actual, es posible que incluso el link sea directo, pueda ser directo, con la respuesta inmune innata a nivel de interferones, pero es muy nuevo. Tengo muchas ganas de explorarlo en algún momento, no tengo las herramientas todavía porque vengo de la biología celular, de la autofagia, de esos tópicos, pero una posibilidad es que hemoxigenasa directamente pueda interactuar con IRF3, por ejemplo, con estos factores que regulan interferones. Y si es así, entonces se podría inducir en forma directa la producción de interferón tipo 1. Así que esa es como mi próxima hipótesis y que me encantaría explorarla.
Any other question? Thank you. The next speaker is Dr. Hernán Peñalosa. He is Assistant Professor, Facultad de Ciencias Biológicas, Facultad de Medicina, Pontificia Universidad Católica de Chile. He is a young investigator of the Millennium Institute on Immunology and Immunotherapy, Santiago, Chile. His talk is Modulatory Role of thrombospondin 1 over neutrophils and macrophages during acute pulmonary Pseudomonas aeruginosa infection. Dr. Peñalosa.
All right. Is it projecting on Teams? We're good? Okay, perfect. So first of all, I would like to thank, of course, the University of South Chile and Dr. Mario Calvo for hosting this incredible seminar. It's been really interesting, all the talks, and also for me it's a privilege to have the chance to show some of the work that we do in Santiago related to, in our lab, related to our interest in immunology and hospital interaction. So just to contextualize a little bit, we all have probably a favorite disease or a disease that we are really interested in studying. In our cases, is pneumonia. The reason why is because it's really important for human burden worldwide. Just in 2019, before the COVID-19 pandemic, it took account 2.5 million of death. And then, if we wanted to find pneumonia, it really affects the alveolar space, which is at the bottom of the respiratory tract. You can see this is the normal anatomy. So you have a epithelial barrier, a very thin epithelial barrier of type 1 pneumocytes, type 2 pneumocytes, and two of them are also very intimately associated with endothelial cells, and this association allows the gas exchange. And also you have some resident macrophages, because this is a tissue or a compartment that might be interacting with the environment, is interacting with the environment all the time. So when you get injury because of pneumonia, for example, you have this barrier disruption, so now the air exchange is not as efficient. You have recruitment of immune cells, and you have edema, so in the worst cases you cannot breathe. Pneumonia is a very heterogeneous disease because it's caused by different pathogens. We are very interested in hospital-acquired pneumonia because it has a very particular characteristic, and that this is caused by bacteria. Antibiotic-resistant bacteria mostly. The risk factors are hospital-associated procedures, such as surgery, for example, endotracheal intubation, that once you are infected with bacteria, bacteria can colonize your respiratory tract, and the mortality is very high, from 20 to 50%. That's why it's really a problem. If you look at the main causes of ventilator-associated pneumonia in Chile, the top 3 causes that took account almost 75% of total causes are caused by 3 bacteria, being Pseudomonas aeruginosa, in 2020 at least, the first cause of ventilator-associated pneumonia. This is not just in Chile. If you look at globally, Pseudomonas aeruginosa is within a particular group of bacteria that causes 13.6%. These are 33 bacteria that causes 13.6% of global deaths. And if you look at closely, Pseudomonas is part of the top 5 bacteria that causes 7% of global deaths by 2019. And most of these diseases, not the only ones, but most of the diseases are lower respiratory tract infections, such as pneumonia. So why Pseudomonas is so important to study? So this is a gram-negative bacterium, and it causes not only acute respiratory infections, but also chronic respiratory infections in some patients with structural lung diseases, such as cystic fibrosis, COPD. It has high levels of antibiotic resistance, can be found all over the world. This is very concerning. And it causes a proteolytic pneumonia characterized by high lung injury. And this is because it is codified for several secretion systems, type 2, type 4, type 6, type 3, that inject virulence factor directly to epithelial and endothelial cells, and also release a lot of toxins, proteases to the environment that causes injury. So we wanted to study the interaction between the host and the bacteria to understand the factors that promote an efficient immune response. So what do we mean with an efficient immune response? When you look at a healthy lung, you see these spaces in white. This is where the air exchange happens, and so the most you have these spaces, the most efficient probably you have the air exchange. So you breathe in oxygen, you breathe out CO2. When you have an infection, bacteria or viral infection, you have an inflammation. This is because, of course, intrinsic virulence factors, but also because an immune response. So you have immune response cells coming into the lungs, and the architecture here changes, so you have less space for air exchange. So what needs to be happened, this response needs to clear the bacteria, but also needs to promote some factors to recover the original structure and physiological function of the lung. So you need to clear the bacteria, but you need to limit your inflammation and your injury at the same time. And this happens due to a dynamic process within this phase of the immune response, characterized by some pro-inflammatory factors that promote inflammation, but also between anti-inflammatory factors that serve as a break of this inflammatory inflammation. And so in our lab, we're basically interested in asking this question to study the host mechanisms that are involved in injury control during lung bacterial infection. And we use Pseudomonas as a model. So which is the molecule that we are studying right now? It's thrombospondin 1 or TSP1. This is a very complex protein, a multi-domain protein that interacts with a lot of ligands involved in different processes, including immunology. So for example, you can see that some domains of TSP1 can interact with neutrophil proteases, or also surface receptors like CD47 or CD36. It is activated TGF-beta. And here I just wanted to show you just how complex this protein is. So not all the protein has been crystallized, but some parts of it we know the structure, and every domain is a different story. So it's very, very complex. It works homoprimer, so this increases the complexity. So why we study TSP1? Because in vivo models using TSP1 deficient mice has shown that these TSP1 deficient mice are way more susceptible to Pseudomonas compared to wild-type mice. This disease model is characterized by impaired lung CFU clearance, bacterial clearance in the lungs, despite have a huge recruitment of neutrophils. And then follow-up works, we have demonstrated that megakaryocyte and platelets that produce TSP1, they essentially modulate neutrophil activation through the inhibition of IL-36 gamma, which is an upstream cytokine that, if not properly regulated, it overactivates neutrophils, and this overactivation process leads to severe injury in the lung, which is not good in this context. So what we did in the lab was trying to understand the factors that break this high pro-inflammatory response mediated by TSP1. So we purchased these mice here, and we brought to Chile, and we repeated the experiment. So here we look at bacterial burden in the lung. As you can see, we replicated what we have found before. So TSP1 deficiency leads to increased bacterial burden at 24 hours. That is not the same at 48 hours. But when we look at other parameters of disease, so for example, this is lung micropermeability, it is high at 24 hours, but it remains high at 48 hours when in thrombus 1 deficiency conditions. When you look at the clinical score, that is how sick the mouse is, it is higher at 24, but it's also higher at 48. And when we look at a physiological protein that we are interested in, which is interleukin 10, we show that at 48 hours, thrombus 1 deficient mice has reduced production of IL-10 in the lungs. Why IL-10 is important, why we are interested in this cytokine, is because what this cytokine does. So IL-10 is an anti-inflammatory cytokine that suppresses a lot of portions of the immune response. It really affects the function of different cells, starting for the myeloid cells, such as macrophages and dendritic cells, but also adaptive cells, such as T cells or B cells, and promotes, of course, the function of regulatory T cells. So IL-10 has been shown to be important in different bacterial models in the lung. So for instance, if there is pneumococcus infection, IL-10 deficient mice are way more susceptible compared to wild-type mice. And this is also shown in Klebsiella pneumonia infection models where IL-10 deficient mice are way more susceptible to wild-type mice. So what happened with Pseudomonas? We asked ourselves that question, and we repeated this phenotype. So just as before, we observed that IL-10 knockout mice are way more susceptible to Pseudomonas compared to wild-type mice. And then we decided to compare the immune response, inflammatory response in these mice. We picked 24 hours because you see there is no difference in survival at this time. And what we found was that these mice have more lung permeability compared to wild-type mice. They are way more sick. The disease severity is higher, and they have problems in controlling the bacteria. So yes, IL-10 is important. It is relevant to control this infection of Pseudomonas. So what is the source? That was the next question we asked. For answer that question, we use a reporter mice that codifies EGFP along with IL-10, and we look at neutrophils. Well, we look at a lot of cells, but I will show you here what neutrophils do. So neutrophils are rapidly recruited to lungs, but then we found that IL-10 positive neutrophils are also rapidly recruited to the lung, reaching at a peak of 48 hours. We quantify this by flow cytometry. Here you can see how at 0 hours there is almost none IL-10 positive neutrophils, but then at 48 hours they significantly recruited to the lung. Neutrophils are very heterogeneous. So we're trying to understand whether this is a subtype of neutrophils or this is a way more heterogeneous population of neutrophils that produce IL-10. We conducted multiparametric flow cytometry analysis. So this is a reduction population of neutrophils. You can see also the neutrophils here in gray. And at baseline, of course, there is a little population of neutrophils in green. These are the IL-10 positive neutrophils that get enriched after infection. But you can see that there are at least 2 clusters here of IL-10 positive neutrophils that are different between themselves. So we quantified them, and we found that almost at least 50% of neutrophils, or 90% actually of neutrophils, were grouped in C1 or C2. And there is just a little, 10% approximately, of neutrophils scattered over this population. So they are at least composed in 2 different clusters. We wanted to study the difference between them, and of course the difference between these remaining IL-10 neutrophils. So compared to other cells, these were the most abundant IL-10 producing cells. You see the neutrophils here, and then all the other cells that produce some degree of IL-10 were at least 1 order of magnitude reduced or lower. So this is not surprising. Actually, it's been previously shown that neutrophils can be plastic cells that can acquire even the classical pro-inflammatory phenotype, but also an anti-inflammatory phenotype. So we wanted to study, ask ourselves the question that we are interested in. So the first question is, well, can TSP1 induce IL-10 directly in these cells? And for this purpose, we need to develop a model. Neutrophils are really hard cells to work with. So we took from mice bone marrow precursors, cells from the bone marrow. We differentiated them with GCSF and MCSF, and as a result, we obtained these CD11B, LY6C, LY6C cells that show an anti-inflammatory phenotype. This has been published before, and this is the phenotype that we obtained. So you see that a lot of these cells express high levels of CD11B in culture, and also high levels of LY6C and LY6G compared to those that are not treated. In terms of how these cells looks like, you have these look like very similar to bone marrow neutrophils. So you have these polymorphonuclear shape cells that resembles really very similar to neutrophils obtained from the bone marrow. So in terms of functionally, we wanted to answer our question. So we took the cells, we incubated when we created increasing concentrations of TSP1. And we found that just leaving these cells with TSP1, which are the red bars here, you see that there is an increase in IL-10 production without any other stimulation. So with 50 micrograms per ml of TSP1, you have a significant production of IL-10. And then when we added LPS, which is a pump that induced IL-10 in these cells, as you can see here, this phenotype is more accentuated. So they work together, TSP1 with LPS, they work together to induce high levels of TSP1, IL-10, I'm sorry. And then when we differentiated these cells from TSP1 deficient mice, we observed something very similar. So wild-type mice stimulated with wild-type cells stimulated with LPS, they produce high amounts of IL-10, but then when these cells were isolated from normal mice, it didn't produce IL-10 as much. So we wanted to have some idea of the mechanism that was leading this production of IL-10 mediated by TSP1. We took these cells, and in the literature you can see that the transcription factor PPAR gamma is very important in inducing this anti-inflammatory program in myeloid cells. So we differentiated these cells, we stimulated with thrombophonin 1, and our hypothesis was that this stimulation was activated PPAR gamma, and then this PPAR gamma mediated IL-10 production. So what we did was we treat these cells with agonist and antagonist of PPAR gamma, and when we look at how the IL-10 production was under LPS stimulation. So when we just added the agonist plus LPS, we have an increase in IL-10 compared to LPS treated with vehicle. That didn't happen with the antagonist, of course, but then when we add thrombophonin 1 to the equation, of course, we have an increase in IL-10, but then TSP1 in presence of the antagonist, this production of IL-10 was gone. So this data actually suggests that this IL-10 production induced by TSP1 is being mediated by PPAR gamma activity. But the next question that we wanted to solve was that whether the reduced levels of IL-10 that we saw in TSP1 deficient mice is actually the cause of this increased or impaired phenotype that we observed when we infect these mice with Pseudomonas aeruginosa. So what we did was we differentiated IL-10 positive and IL-10 negative cells, and then we transferred them intratracheally to TSP1 deficient mice. And then one day after transfer, we infect these mice and see how was the lung inflammation and the host defense. And we observed something very interesting things, like for example, compared to controls, this is the TSP1 knockout mice that were transferred with PBS, with this vehicle. You see that there is a recovery and increased production of IL-10 that was kind of comparable with the control mice. And then when we look at the bacterial burden in the lungs, we see that the transfer of TSP1 deficient mice with IL-10 positive cells, it really reduced the bacterial burden. It really reached to similar levels compared to the control. And then the clinical score was also reduced. So disease severity, these mice looks much better when you just look at them after infection. The lung permeability also went down to equivalent levels as wild-type mice. And then we observed that there was a decrease in neutrophil recruitment. So this data actually help us to establish that TSP1 does not only modulate the pro-inflammatory function of neutrophils as was previously shown, but it also produces or enhances the phenotype of anti-inflammatory phenotype in these cells. That is mediated by a production of IL-10 through PPAR gamma activity. And this phenotype or this function, this neutrophil function basically helps to mediate protection of the lung tissue during infection. Now, neutrophils are not just the only important cells in the lungs. There is a lot of data that shows that TSP1 can somehow affect the function of alveolar macrophages. And in different phenotypes, in different models, this is an LPS in this model. So we asked ourselves whether TSP1 can modulate the alveolar macrophage function. So the first thing we did using the same mouse model, we quantified the cells, and in both cases, in the wild type or knockout, we observed that there is a decrease in the alveolar, in the resident macrophages in the lungs. There was no difference, so we decided to go deep further into the function of these cells, and we decided to do it by a multi-parametric flow cytometry. So these are alveolar macrophages at 0 hours between wild type and responding knockout mice, 24 hours and 48 hours post-infection. You see the decrease in the percentage of this population. But you see that there are some differences here at 48 hours between wild type and responding knockout mice. What you also can see is that these cells are very heterogeneous. So there is not not all the alveolar macrophages are actually doing the same or they have the same phenotype. So they differently express several markers that are involved in early immune response. So we wanted to study how these cells are changing and what does that mean. So we run a phenograph analysis. We separate these alveolar macrophages in 17 different clusters. You can see that here, each one of these clusters express each marker differently, so they are very heterogeneous cells. And I think that's one of the messages that I wanted to give you. And we evaluated how this population changes over time in either wild type or TSP1 deficient mice. So you can see that there are at least 3 clusters that changes over time. So cluster 3, they got amplified even in wild type or knockout mice over the first 40 hours. And then you have cluster 12 that it really disappears after infection in wild type mice and also in knockout mice. And same with cluster 14 that it is at baseline, but then it's gone. And we also wanted to evaluate whether there was a difference between wild type and TSP1 knockout macrophages. And we found some. So for example, cluster 7 here is over-amplified at 24 hours. Based on the expression of a surface marker, we classify them as inflammatory macrophages. And these are way more pronounced in wild-type mice. And then same with cluster 9, they are more pronounced in wild-type mice compared to knockout mice. So we're trying to figure out what does that mean in terms of functionality. And also we are trying to extrapolate this data to other subtypes of macrophages either insert tissue macrophages or monosaccharide macrophages. So overall, our aim is to try to identify immunomodulatory compounds such as TSP1 or cells such as IL-10 producing cells or IL-10 producing neutrophils to try to somehow use it as a potential therapy. You know, the mortality caused by these infections are really high, so we plan to, or our hope is to develop these tools to reduce the patient mortality, of course, to reduce antibiotic usage, reduce the spread of antibiotic genes involving antibiotic resistance, and of course, reduce the transmission of antimicrobial resistant bacteria across the world. So, well, this is my lab. The people who actually do the experiment, I would like to acknowledge or say thanks to the funding sources, and of course the collaborators in Chile and also abroad. Thank you.
Any questions? We have time for one question.
I'll try to be a good question. Yes. So you showed the differences in the populations between the wild type and the transponding knockout mouse, and they were quite similar, except the population you made a sub-analysis, right? So do you think those cells are the main responsible for the phenotypes observed in the other experiments, where you focus on those cells, or do you think it's just a bystander effect?
So the reason why we wanted to study, you mean macrophages, right?
Yes.
So the reason why we wanted to study alveolar macrophages is because they are the first immune cells that get in touch with the bacteria during infection. So,
Sorry, that phenotype, the phenotype of those C7, that
yes, so we wanted to, our hypothesis is that that's the phenotype that we need to address. So that right now we are doing some functional assays. So you isolate neutrophil macrophages, you incubate them in vitro and see some. We are trying to ask that question using that model. Focus on your question.
And how will you be able to target them only compared to the other cells?
Yeah, so we are not trying. So the approach that we are trying to take, because this is so hard to sort cells, population specific cell populations, is try to find a function that is different between in presence of TSP1 and absence of TSP1, and see whether or not that has an effect in bacterial clearance of host defense.
So RNA-seq, probably single cell RNA-seq,
For example.
Yeah.
Thank you, Dr. Peñalosa. Now we need to run because we have only 10 minutes for the coffee.
adults, we have at least 2 times more hospitalizations and even 20 to 100 times more death. So it's a huge burden in the older age. And we have every time older people, and we have more comorbidities, and therefore we have a higher risk of being hospitalized because of RSV. This is an interesting point too, because we have very present influenza, and even in Chilean population we know influenza vaccine, and that's something that happens every year. But this is a comparison, a study where they compare people who was hospitalized before RSV disease or influenza. And with RSV, you have a longer hospital stay, and a hospital stay of 7 days or more is very high impact for our health system. You have more pneumonia, more ICU admission, more COPD exacerbation, and even more mortality. And in this part, they compare these 2 cohorts of influenza and RSV for 1 year. And you have more risk to die because of RSV than for influenza infection after 1 year of your hospitalization. So it's really relevant. And for the health outcomes, we have RSV is associated also with big issues when you have the infection in immunocompromised populations. More affect are the allogenic hematopoietic stem cells transplantation, because you don't have the right immune system to defend yourself from the infection. But for lung transplant recipients, one part is what the virus do in the lung itself, in the tissue, but also the RSV has a lot of impact in immunologic responses. So you have even the risk to, in 1 of every 3 people who have a lung transplant and get infected with RSV, you have the risk for chronic rejection of the allograft. So it's a huge impact for these patients. Other patients at risk with solid organ transplant or leukemia, lymphoma, tumors, or immunosuppressive therapies that they use for chronic these conditions, you have 1 of 4 of these patients will be hospitalized if they get infected. So also what we know from before, we have exacerbations like from COPD and asthma. For asthma, you have an exacerbation and you get better, but for COPD, you have a decline of lung function. And these patients will not recover their initial lung function after every hit, and in this case, with RSV infection. The other part, and RSV is acute respiratory infection, but you have an all-cause increase in mortality and also myocardial infarction risk, heart failure, and risk for hospitalization because of the disease. And one point that it's well known for influenza, for example, and now we have much more information for RSV than before, is that the risk of MACE. MACE is major adverse cardiovascular events like myocardial infarction, heart failure, but also includes arrhythmia, venous thrombosis. If you have diabetes or any other comorbidity, you have much more risk of these events if you get an RSV infection. And we have long-term effect. One is the frailty or functional decline. Frailty is the ability, or that you have, it's for older adults that you can have more complications, and not just functional decline, also cognitive impairment. And the third one is what we're going to talk a little bit more, the cardiovascular outcomes. So for frailty, if a person gets infected, in this study they show what happened after an infection who needs an hospitalization for RSV, and 1 of 3 had a sustained functional decline. So you cannot do the same thing that you did before your hospitalization. That's a huge impact in your normal life. 13% of the patients needed to go to, for example, nursing home or somewhere where they needed more care for daily living. 20% had cardiovascular events or heart failure. 80% had a decrease of the lung function, especially because of COPD disease, and 38% needed a re-hospitalization after this event. So it's a huge impact. It's not just one hospitalization, you have more risk of getting 2 times to the hospital and more complications. For the cognitive impairment, we saw in the last talk the huge impact of pneumonia, and there are some studies that look at what happens if you get pneumonia and you have to get hospitalized. Even if you don't have any impairment before, there is a risk that you have after pneumonia impairments of activities for daily living. Really like to have independent daily activities. So it's a huge impact for each patient. And for RSV, if you get an older patients, almost 27% of the patients who get infected will develop pneumonia, so are at risk of this cognitive impairment. And the other part is brain fog. If you get infected by a virus, normally you feel that you don't feel well, but sometimes you really have the feeling you don't think well. And that's just a really, it's a true thing. It's not that something that you have the feeling. It's direct viral neuroinvasion. This has been demonstrated for RSV, also for influenza, neuroinflammation. And if you get a pneumonia, you also have issues with oxygenation and neural function. So brain fog is a really issue, and this can even not be just in the infection itself. It can last for months or weeks and months. So the cardiovascular outcomes that I mentioned before, this is a study with almost 12,000 patients. And they look at the risk for cardiovascular events after an RSV-related hospitalization. And it's a self-controlled US database. So you see if you have the infection and a risk period that they evaluated for until 180 days. And here I want to show you that you have more risk, and especially in the 2 and 3 first weeks, for have a myocardial infarction, for have heart failure, to have stroke. But interestingly, even for stroke, you have more risk until 180 days after your infection. So this is not just the first hit, this is a prolonged term sequela of the first infection. Other severe health outcomes because of RSV, this is if you don't have, you cannot see. So if you don't have RSV, the risk for any cardiovascular event is much less than if you have RSV, and this can be until a year. So it's not just 180 days, until a year after your infection. If this is a severe infection, you will have issues after it. And this 1 year risk of cardiovascular events is comparable to what influenza infection does in the patient. So as we have more conscious about influenza, we should have it with RSV too. In this, what are the most important cardiovascular events is that you have an exacerbation of heart failure, that it's the more important consequences of the RSV infection, but also you may have acute myocardial infarction. And to have an infarction, it could be something that you can treat in the one hospitalization, but you can have heart failure after it, or subsequent problems with coronary heart disease. And this is something that first was studied, as I mentioned before, with influenza. And you have a lot of cytokine release and inflammation. The macrophage that we talked before in your alveolum is going to be activated. Every infection, even bacterial infection, may rupture the atherosclerotic plaque and may lead to thrombosis, and then you have your myocardial infarction. But also, if you have a pneumonia, you will have, because of, for example, these 2 viruses, you have a lot of inflammation because of cytokine release, but also because there's the lung, the tissue that is involved, you will have hypoxia, you will have tachycardia, sympathetic activation, vasoconstriction, and then the different cardiovascular outcomes, with every of them risk of morbidity and mortality. For RSV, this is a huge cytokine release, especially if you have low respiratory tract disease, and that was one of the main points what vaccine prevention was studying. So you have liberation of interleukin-6, interleukin-1b, TNF-alpha, with endothelial dysfunction after it, increase of vascular permeability, and this prothrombotic state is for the coronary artery disease relevant, but even the cause of prothrombotic effects for example, deep venous thrombosis of your legs or even pulmonary embolism. Then, if you have pneumonia, you may have a hypoxemia, you have a cardiac overload, and then you have an acute discompensation that can occur for the first time, or if the person has a heart failure before, it got an exacerbation, and then we get this long hospitalization for more than 7 days, and the whole health cost involved with it too. With sympathetic activation, there will come the demand ischemia and also the arrhythmias, very frequent with low respiratory tract infection with viruses. For RSV prevention, in the beginning we have especially monoclonal for the infants, and then we got the vaccine approved for the FDA. The pivotal studies were May 2023 for the adjuvant vaccine, and in the same month, a little later, with the Renoir study with the bivalent vaccine in adults. Then we have the approval for the maternal vaccination. There the intent is to vaccinate the mom, to protect the infant until 6 months, to avoid a lot of the morbidity and mortality in this population. And then we got the approval for the mRNA vaccine, that we don't have in Chile, but we have for the 2 vaccines before, with the adjuvant and the bivalent in Chile. So in maternal vaccination, we have the bivalent vaccine, and for older adults or adults with comorbidities, we have both recombinant vaccines. And well, we talked a little bit now with the approval of these vaccines, and they have efficacy in the RCTs where there was for the first studies. But normally in these studies, the immunocompromised patients were excluded. In the real-world evidence, we had a very broad population of patients that are included there. People over 80 years are less than 80% of the participants of the first RCTs. There are more, 1 or 4 patients are at this age, but they studied in real-world evidence. For chronic conditions, if you have patients over 60 or 65 years, have just 50% of comorbidities, it's because of the first studies. In the real-world study, we have much more comorbidities studied before. And in the approval studies, we have symptomatics and especially low respiratory tract disease, because it's an event that is more easy to study, and you need to show the impact in the first beginning. But if you want to have real-world evidence, we need to know how many emergency department visits can we avoid. How many hospitalizations do we avoid? Have you ECO patients, or can we prevent death? Can we prevent other RSV-associated health outcomes? So this is what the real-world evidence confirmed until now. I will show you one of the studies. You see the number, this one is not so high, but the other 3 studies that I included in the evidence before are the third one, even with 150,000 patients vaccinated and their outcomes. They are similar. There are studies from real-world evidence with the first season after the approval of these vaccines. In this study, we have 24% of immunocompromised patients, 96% of the patients have at least 1 chronic condition, and this is a study conducted from the CDC, so they were included both vaccines they had in the moment, the adjuvanted vaccine and the bivalent vaccine. So, and the results are that it's very similar to the approval study, so almost 80% of effectiveness to prevent hospitalization due to RSV in people with 60 years or older. And that's a very good result for this real-world evidence. And this is a very interesting study. We, with our colleagues, assisted to RSVNet. This was in February in Italy this year. And the DAN RSV study was a RCT done in Danish population. The most of the people were 69 years mean. And they studied the bivalent vaccine. So they invited people to get vaccinated, and the controls were people with the same age and conditions without vaccination. And the interesting part, and there was a very broad event what they studied, they just focused in something what was hospitalization for any cardiorespiratory disease, not just like the lung infection because of RSVs, no, any cardiorespiratory disease as outcome. And chronic disease in this population was 40%, that's acceptable, but if you see a little bit more, chronic lung disease, just 7% in this whole population, diabetes just 11%. At least in Chile we have at least between 30 and 40% of the population in this age with diabetes. So, or for chronic cardiovascular disease, just 20%. This was very healthy people that got vaccinated. And that's important to consider because the outcome of the study was this was a really huge difference in hospitalization for cardiorespiratory disease in this population, even if there was very healthy people, were very healthy people. And for the events of cardiorespiratory disease for any cause, for the hospitalization, they have 26 events in the vaccinated group, 29 in the control group. This would be a absolute rate reduction is just 2.9, but there's still 10% reduction of risk for hospitalization in this population that were very healthy. I have to repeat that again because if you use the same data for people who with work comorbidities, you should suspect that we will have a bigger result. So this suggests that we can, with immunization for RSV, prevent all-cause cardiovascular hospitalization. And there was, after this, and this was an RCT very well done and controlled in this healthy population, but we have also, they presented poster, for example, in ResVinet, they calculated in the same Danish population how many people you need to vaccinate to prevent one cardiovascular event with the need for hospitalization. They need 344 vaccinated people. So that is not a big number of people you need to vaccinate to avoid these outcomes that could compromise your life. So that's why I wanted to share it with you today. We have a lot of knowledge that we still need to address, especially because how these different viral infections go to so many cardiovascular events. And if we could address that better, but now even in the guidelines we have vaccination should be part of the cardiology management of our patients. So thank you very much. If there is any question.
So, are there any parameters that it would be noteworthy to deepen on the type of immune response induced by the vaccine between individuals that might separate those that are protected from cardiovascular disease from those that are not, that you would look at, for example?
The problem is we don't know exactly which part of the immune system is different in that part, and we know that for the vaccine studies, immunosuppressed or immunocompromised patients have less vaccine effectiveness they have more viral infections. And it's a difference even for the second season, for example, from 70 or 60% it go down to 40%, so they may even need more vaccination. But this impact of the viral infection in patients, they could have no comorbidities before and still have a cardiovascular event. So normally in vaccine studies you look for antibodies, you look eventually for cellular response, but there is still a lack of knowledge between your immune response and your like what is going to happen in your atherosclerotic plaque. So there is where we need more information, so which is the part what we could change, because we're using aspirin now. This is very broad type of, and we know that still using aspirin, the people still get myocardial infections. So now we know that you should reduce the infections, but which part of this immune response is really involved is no questions.
Okay, thank you.
Thank you, Dr. The next speaker is Dr. Leandro Carreño. Dr. Leandro Carreño, he's Associate Professor of the Facultad de Medicina, Universidad de Chile, Deputy Director of the Millennium Institute on Immunology and Immunotherapy, Santiago, Chile. And his talk is Modulation of B-cell immunity by natural killer T-cells.
It's going okay in the teams, Pablo? Fernand? Is okay in the okay? Good. So thank you very much to Mario and people here at the Universidad Austral. It's always a pleasure to be here, and also the organizers of this symposium. Thank you, Alessandro, for coming here, so far and be here until almost the end. So I'm going to share today something a little more. We're going to go back to the basics, and I'm going to, let me see if this should work. Yeah, it's working. Let me just adjust my hand. Okay, good. About B-cell immunity, and I think, because we have learned, today how important are antibodies, so I'm going to show you some work that we're doing in the lab, trying to modulate, fine-tuning the B-cell responses using these cells that we are interested in the lab, which are natural killer T-cells. So let me just start with a basic overview of the immune system, just something that we have been followed in the previous presentations, and just a reminder that the immune response starts with recognition of some danger signals. You call it the pathogen-associated molecular patterns, or sometimes there are actually danger signals like DAMs, etc., that will enhance the activity and will allow the dendritic cells to become mature and then migrate to the lymph nodes and activate the T cells, CD8 T cells, CD4 T cells. Because they will present the antigen that is derived from the pathogen, but also the second signal, which is the co-stimulatory molecules. So that will allow the stimulation of the CD8 cells and the CD4 T cells. CD8 T cells, which are cytotoxic, can actually mediate the direct killing of infected cells. For example, in this case, in this cartoon, there is a bacterial infected cells. It could be a virus as well. But also, the CD4 T cells, they will enhance the response of other cells, such as B cells, which at the same time, will recognize via the BCR, the B cell receptor, another part of the antigen. In this case, it's a bacterial antigen. And because the T cell has been activated with the same pathogen, so to recognize also bacterial-derived antigens, they both actually recognize the same antigen specificity, different epitopes, but the same antigen, the whole pathogen. And that will allow the differentiation to plasma cells, and then the production of antibodies. Antibodies will block the pathogen, and that's, a productive immune response. So this, unfortunately, doesn't work all the time. Sometimes we have pathogens that can invade the immune system, they can escape, they have mechanisms that will actually mediate an immune response that actually are not good or detrimental for the host. And so we are always looking to have different you know, different mechanisms or treatments, you know, to redirect, to train immunity, specifically to enhance the CD8 T cell responses, the CD4 T cell responses, and also the B cell responses. So there are other cells that actually take part in this initiation of the immune responses, which are mediated by dendritic cells, which are the ILC cells, the innate lymphoid cells, which are three types, ILC1, 2, and 3, because they actually have the counterpart of the Th1, Th2, and Th17 T cells, CD4 T cells, but also NK cells, macrophages, and NKT cells. And in my lab, we are actually very interested in these cells because they have a very important feature. We can target them with some ligands, and I will show you this in this slide. So they're called natural killer T cells because actually they are T cells. They have a receptor, a T cell receptor, you know, the same that harbors a T cell, a conventional T cell, like a CD4 or CD8 T cell. This T cell receptor has two chains, an alpha chain and a beta chain. But the difference between the both receptors, you know, in a T cell and in an NKT cell, is that this receptor is semi-conserved. So the alpha chain is actually always the same. The beta chain has limited repertoire. And also, they express some receptors that are commonly found in NK cells. So they can have intrinsic killing properties, recognizing the balance between activatory or inhibitory signals. They are T cells, they derive from the thymus, and they communicate with other cells in an innate immunity scenario, so they can actually mediate the enhancement of the activity of the dendritic cells, B cells, CD4 T cells, CD8 T cells, and NK cells, just a little bit before the priming of the dendritic cells to T cells. So how they can do this? Because of the TCR. The TCR recognizes a glycolipid that is actually bound to another molecule, which is a MHC class 1-like molecule, which is called CD1D. In contrast to what happened with the MHC class 1, this molecule is extremely conserved. So actually, most of us have the same sequence. They have very limited differences, and actually even conserved in different species. So the NKT cells, once they recognize the ligand, they can secrete different and a large amount of different types of cytokines, including interferon gamma, IL-4, but also IL-13, IL-13, even interleukin 10. So the most studied ligand, which is here, we'll just zoom in here. So this small molecule is a glycolipid that is bound to this molecule. So we can see here the structure. This is the most used glycolipid, which has identified in a marine sponge several years ago. The name is an alpha-galactosyl ceramide, so we have a galactosyl part of the molecule here with an O-glycoside linkage and a very large acid chain and a sphingosine. So this compound was identified almost 25 years ago, and it was proven to activate most NKT cells in both humans and mice. And because you know of the interest of having a distinct cytokine signature of the different NKT cells, there has made many modifications on the chemical structure of this molecule. We have here, some of them are very subtle, like here, this O-glycoside linkage is replaced by a carbon. In this case, this molecule have a functional group in the acyl chain. This here, it has a shortened version of the acyl chain. This one has some insaturation, etc. So the different chemical modifications of the compound, it has become very useful to identify and characterize some glycolipids that have a more distinct Th1 biasing or Th2 biasing. But 30 years ago, I think, not even 10 years ago, I think, this has been established almost 6 to 8 years ago, that the NKT cells, actually this has been established by our group and other groups, of course, in our lab, that the NKT cells can also adopt different phenotypes, like, it's very similar to what happened with CD4 T helper cells, and also with ILC cells, that we call NKT1, NKT2, NKT17, and even NKT10 cells. They have different, as well as the counterpart, they have actually signature distinct transcription factors. They secrete different cytokines. And we were very interested in this year to set ways to identify them and also try to direct the response to have a unique phenotype among them using different glycolipids. So just for reason of time, I will skip some part of the identification of the glycolipids, but I can say that we have identified different glycolipids, and this is a good thing about NKT cells because they can be specifically targeted by these different glycolipids, and they can acquire a pro-inflammatory phenotype that is useful for infections and cancer, or an anti-inflammatory phenotype that can be useful for allergies and autoimmunity. So just a quick example. Here you have a mouse experiment where you have a BCG and also a modified version of BCG with a pro-inflammatory glycolipid. So the BCG will, of course, lead to the generation of antigen-specific CD4 and CD8 T cells. You can interrogate them with the re-stimulation with TB antigens. Remember, the BCG is the current vaccine against TB. It's not that good, but we can discuss that later in the coffee. And you can see a specific T-cell response here. This is an ELISpot, interferon gamma ELISpot of CD8 T cells. And that response is correlated with the protection of the infection. If you have, at the same time, the NKT cell activation, because you have glycolipids into the bacteria, in the BCG itself, this NKT cell activation will lead to an enhancement of this response that you can see here, and also a better protection. This is the same when you have a model, for example, of melanoma, when you have a metastasis in the lungs here, you previously injected some glycolipid and you can see that the response it's better. And you can also even identify some glycolipids that are better than others. So this is just to show you the scenario. We're very interested in set up different glycolipids that can go to different ways. And we study the biology of the NKT cells, the structure function of the ligands and receptors. This is some part that I did out of this presentation, how this can be useful for immunotherapeutic potential in different models. And actually, I do collaborate a lot with the colleagues here in this room. But today I will talk to you about the costal with other immune cells, and specifically B cells. And I think it's very important to this symposium because of the importance of antibodies that we have seen. So NKT cells can interact with B cells because B cells also express CD1D and can present a glycolipid. So the current view of the B cell responses, as you can remember from books and from basic immunology, is that you have an antigen, the dendritic cell will lead to the activation of CD4 T cell, and at the same time you have the same antigen with a BCR epitope. Then you have the communication between the CD4 T cell and the B cell, and that can lead to the expansion of short-lived plasma cell, the not so good antibodies, but also to the germinal center reaction, which will lead to the memory cell formation, the isolate switching, the affinity maturation. So we were very interested to see if we can surrogate the CD4 T cells using NKT cells here. And since we have been identifying in the lab different glycolipids that have, lead to different phenotypes of the NKTs itself, like pro-inflammatory, anti-inflammatory, so we did those experiments. I'm sorry. In order to have the delivery of the B cell antigens and the NKT cell antigens, we use nanoparticles, liposomes, as you can see here. In the liposome, you can have the antigen
inside the particle or outside the particle. In this case, I will show you outside the particle, and embedded in the membrane, the glycolipid. So this is the size of the glycolipid. This is just an equipment that, you know, with the flow you can, and the electricity, the conductivity of the electricity, you can actually measure the diameter. This is an atomic force image of the liposomes. There's some quality control things that you have to do in order they have to be in a range of 100 to 200 nanometers to be acceptable for pharmaceutical industry. So we're there. We can measure the conductivity. We can see them in electronic microscopy. We can put the antigen here, and you can see the antigen by flow cytometry in the liposome. With those liposomes, we immunize mice, and we interrogate how is the B-cell responses. We use different type of glycolipids. The prototypic one that is very pro-inflammatory, and one other that is anti-inflammatory. As you can see here, that the glycolipids itself, they actually enhance the response of the antigen, which is OVA in this case, but also they change the composition of the different isotypes, which is very interesting for us. For example, in this case, this one is Chile2026 is the name. This modification is very important for us because we are pursuing to be an excellent candidate for antiviral responses. They have a more IgG2B, for example, isotype, but they have more relative ability itself. So we also interrogate the cells. So this is flow cytometry for specific OVA antigen cells, and you can see that this antibody production correlates with the expansion of the cells that are positive for IgG2C and IgG3 and IgG1 in the case. So just to finalize the story, we were interested to see whether this NKT cell surrogation can also affect the expansion of T follicular helper cells and germinal center B cell, which are important for the memory establishment and the high affinity antibodies. We could see that when we compare even with the normal adjuvants, we see a better effect when we use these two glycolipids here, the alpha-galcer and the histine-7. So we are very actually excited to see about this. And this also correlates in an experiment when we follow the long-term immunity. I mean, this is mice. I know that the 81 maybe is not a lot, but for mice is a lot. After a prime boost regime, you can see with these two glycolipids, this is flow cytometry for the switch B memory cells, and also the antibody secreting cell from the bone marrow. This is a specific ELISpot. So just very short, we also like to see if we can see an effect of the T-independent responses, because it's reported not to be, not to generate a lot of isotype switching, and we were very actually happy to see that also the immunization with T-independent antigens, in addition in liposomes to the glycolipids, can enhance the isotype switching for IgG1 and IgG3 in some T-independent antigens. This is very preliminary. We are working on preparation of this, and this is actually correlated because we're very interested to see if this is because of the germinal center or the specific actually T-independent cells. So we follow them by flow, and you can see that they are the marginal cells, B cells, that actually changing the isotype to IgG1 and IgG2. And the same happened with plasma cells, although we are not very sure that this is exclusively for marginal soluble cells, so we're doing more experiments. So just to conclude on some questions that are open right now, we could see that NKT cell activation by different glycolipids could lead to a difference in isotype switching, this fine-tuning that we saw in one of the slides. And we still want to know if it is possible to have this difference using different priming regimens. the quality of the T cell memory, how that is different, and also can this result be translated in a disease infection setup. And for this, actually, we have planned some experiment with Alexis and Pablo. And this is also, kind of exciting for us, the T cell independent response. So we wanted to know if B cell memory is also present. And this result be translated in a bacterial infection setup. We're collaborating with Lisan Pirovsky right now in Albert College of Medicine in a model of a bacterial infection. So with this, just to like to acknowledge the natural killer tesistas, which in the lab here, former members, a large group of collaborators, very fond of them, that made this possible. Of course, the funding. Thank you very much. I'm happy to take questions.
Any question for Dr. Carreño? Any question? Well, thank you.
The coffee.
Our last speaker will be present online. Then we need to connect.
Can you hear us?
Now I can hear you. Hi, good afternoon. Can you hear me?
Yes.
Can you share your screen?
Yes, I can hear you right now. Okay, can you hear me?
Yes, we can.
I guess you can put it in the chat as well.
I will introduce you. I'm Dr. Mario Calvo. Nice to meet you. Our last speaker is Dr.
Unfortunately, it comes and goes. I cannot hear the room right now, but I'm hoping that you can hear me.
We can listen to you. We can listen to you, but we will turn off the microphone.
Okay, excellent. Then probably I'm good to go ahead. Is that correct?
I'm sorry?
Do you want me to start?
No, I will introduce you first. After that, I will turn off the microphone.
Sounds good. Thank you.
Our last speaker is Dr. Rassam Erdem. She's Global Clinical Lead and Director in Vaccine Clinical Research and Development at Pfizer, PATH, and the U.S. National Institute of Health in the United States. She will talk about results from a trial of RSV-pre-F vaccines in pregnant participants living with HIV in South Africa. Please, Dr. Erdem.
Thank you so much. Good afternoon, everyone. I hope you can hear me clear and loud. Thank you for the invitation and for the opportunity to talk to you and present you this data today. And I hope you can also see my slides. Today I will briefly cover the unmet medical need, summarize our Marisol design, then walk you through the maternal and the infant safety, and finally the exploratory immunogenicity comparison to Matisse trial from our South African HIV positive mothers. Today, my presentation is the results from a trial of RSV pre-F vaccination in pregnant participants living with HIV in South Africa. This study is called C367-1032. It's a phase 3 randomized placebo-controlled trial evaluating the safety, tolerability, and immunogenicity in mothers living with HIV and their infants. This was a phase 3 trial. We conducted it in multi-centers within South Africa. It was a randomized, double-blind, placebo-controlled study in this particular country. Country selection for this trial was critically important because in our Matisse phase 3 trial, which was our pivotal trial for the licensure of RSV pre-F or Abrysvo, we detected some numerical, non-statistically significant imbalance in the preterm rate in South Africa, which was a topic of concern from many aspects, including regulatory, and this trial became more critical to evaluate the same population and HIV positive mothers, which is a significant number of mothers within South Africa. We assess safety, tolerability, and immunogenicity of the Pfizer's RSV pre-F in pregnant participants living with HIV and their infants. The study enrolled 343 pregnant women living with HIV, and we vaccinated them between 24 and 36 weeks of gestation. They were randomized 1-to-1 to RSV pre-F or placebo, and the eligibility required stable HIV disease before randomization. And the determination of the eligibility key criteria was based on the locally accepted guidelines. That included viral load being less than 1,000 copies per milliliter, and the CD4 count being more than 200 cells per millimeter cube.
We have a problem with the laptop, and we will check the laptop. Only 1 minute, please.
Sure, absolutely, I will wait.
Sorry.
No problem.
you can return. Thank you. Now you can start again. Yes.
Okay. You want to start from the beginning or continue where I was?
Just a minute, and we will let you know if we can start discussing this slide again. Sorry.
Okay. Okay. I think we're okay to start the slide. This is the interaction after all.
Now we're going to start again. Thank you so much, and sorry for the inconvenience.
No problem. Thank you. All right, let me introduce you to our trial. This is C367-1032 Marisot trial. And if you look at the study design at a glance, this was a phase 3, multi-center, randomized, double-blind, and placebo-controlled study in South Africa. The location of the country was critical due to earlier signal, preterm birth signal in South Africa in our phase 3 Matisse trial, which we did not duplicate or see the same signal in this trial, which I will present you today at a later slide. We assess safety, tolerability, and the immunogenicity of the Pfizer's RSV-PF in pregnant participants living with HIV and their infants. The study enrolled 343 pregnant women living with HIV. They've been vaccinated between gestational age of 24 to 36 weeks in this country. Participants were randomized in 1-to-1 ratio, 120 microgram RSV-PF or placebo. Eligibility requires stable HIV disease before randomization, and we described the stable disease based on the South African local guidelines, which included key criteria of viral load being less than 1,000 copies per milliliter and CD4 count being more than 200 cells per milliliter cube. Participants were also on the stable HAART regimen for more than 90 days, and we did collect blood samples from mothers and the infants as well. So when we look at the safety, we included reactogenicity, AEs, severe SAEs, AESIs, and the newly diagnosed chronic medical conditions. And immunogenicity is presented descriptively, including comparison to non-HIV. During pregnancy and postpartum. And HIV-exposed uninfected infants also have higher hospitalization and death rates from RSV, LRTI than the HIV-unexposed infants. And these burdens are also supported by many internal and external publications as well. You can see the list at the bottom. If you look at the study schema, let me orient you what we did in this trial. Maternal cohort, the pregnant participants were younger than 49 years of age. And they were with well-controlled HIV. We started with 433 participants. The visits were really well structured. The day 1 being visit 1 vaccination visit. Visit 2 occurred 1 month after vaccination. And we had their delivery visit, which is also accompanied by a blood collection. And the last visit was 6 months post-delivery, which was the end of the study for the mom, for the maternal follow-up of 6 months postpartum. And if you look at the infants, which I will show you in the next slide, which is pretty similar to the maternal participants, but in this slide also I want to refer you to the allocation. It was 1-to-1, and the comparator was placebo, and it was on the same schedule. We collected reactogenicity by e-diary, and also additionally AESIs and the SAEs were collected during the same window. You can see the ESIs and SAEs were throughout the study for 6 months for the moms. The definition of AEs are essentially I think one important thing I should emphasize here is the well-controlled HIV definition. Again, less than 1,000 copies, more than 200 cells for CD4, and more than 90 days. And the good part of this study is that majority of over 80% of our participants were maintained their well-controlled HIV status by the time of delivery, which was a positive finding for us in this trial. So if I move to the infant participants, we had 337 infant participants in our evaluable population, and they had their first visit at the time of birth. We collected their cord blood, which was a preferred method of blood collection for this visit, and infant's second visit was 1 month after birth, and the last visit was 6 months after birth, which was the end of the follow-up for the babies. And at birth, infants had cord blood draw and AEs, AESIs, as well as SAEs collected either up to 1 month or the 6 months, depending on the type of the adverse event we are evaluating. So I would like to focus you on this slide of maternal demographic. On a positive note, they were very well balanced and similar across the RSV-PF and the placebo groups. Demographics are shown for both populations, and as you can see, it was 172 participants on the RSV-PF arm and 170 participants on the placebo arm, of total of 342 participants evaluable for this demographic data. Median age was 31.3, and for the RSV-PF it was 31-year-old, placebo 31.1, and the median age was around 31 for these mothers. And gestational age was important for us. Gestational age at vaccination was 29.5, and the median gestational age was also approximately 29.3 for both placebo and RSV-PF mothers. And based on our Matisse data, we looked at the breakdown of the moms at the time of the vaccination, what their gestational age was. And the study team, during the conduct of the study, put extra effort to make sure that this breakdown from 24 to 28 weeks, 28 to 32 weeks, and 32 to 36 weeks were well balanced and equally distributed between the window of the gestational age at the time of vaccination. And as you can see, approximately 32 to 35% of the participants equally divided between those windows of vaccination by gestational age. In this slide, we are looking at the maternal participants, those who maintain their undetectable HIV viral load and CD4 count status throughout the delivery. I'm not going to repeat the criteria, and but the one thing that's important, this criteria was based on within the 14 days prior to vaccination, and it is determined in collaboration with South African Department of Health 2023 antiretroviral treatment guidelines, with 2 consecutive HIV viral load being more than 1,000 copies, which qualified as a treatment failure, and those were excluded from the study. So if you look at this table, you will see that it's breakdown of the HIV RNA, more than 1,000 copies, versus CD4 count being less than 200 cells. We had, at the time of vaccination, just 1 participant, and time of delivery, 9 participants in the RSV pre-F arm, and 7 participants in the placebo arm were less than 200 cells, and 5 participants had more than, in the RSV pre-F arm, more than 1,000 copies, and 2 participants in the RSV pre-F had more than 1,000 copies at the time of delivery. But other than that, approximately 80% of the participants maintained their undetectable status of HIV at the time of delivery, and 81% maintained that at the time of vaccination. Just based on our Matisse outcomes, we did develop additional tables on this pregnancy outcomes. They were similar across the RSV pre-F and the placebo group, and we looked at the median age, median time from vaccination to delivery, which was important because the time to vaccination to delivery was a big determinant of the immunogenicity assessment and the outcomes in our Matisse trial. It made a big difference when the time from vaccination to delivery was less than 14 days. That was a significantly, statistically significant difference from the other mothers who had more time after the vaccination till delivery. We had only 3 stillbirth as a pregnancy outcome. Unfortunately, 3 events of stillbirth in this population. And gestational age at delivery, median GA at delivery was 39.3 weeks, because majority of our babies were full-term babies. And the mode of delivery breakdown was between vaginal cesarean section, and the C-section has been subcategorized as elective, semi-elective, and emergency, and only 20% of approximately 20% of these C-sections were emergency. Remaining C-sections were either elective or semi-elective in nature. After this, now I will move into the primary endpoints, and I will start with the safety data. Let's look at the solicited local reactions in mothers. This is the maternal data. They were mostly mild to moderate, and they resolved quickly. This chart shows erythema and redness, induration and swelling, and the injection site pain by maximum severity. And this is within the 7 days after vaccination, and it's comparing the RSV pre-F to placebo. As you can see, specific percentages are displayed here. The most prominent reaction was pain at the injection site, but again, let me emphasize, all these events are mostly mild and moderate, and they resolved anywhere between 1 to 3 days in time. If I move to the next slide, now we are looking at the solicited systemic events in our maternal participants. Similar to local events, systemic events were also mild to moderate, and they did resolve quickly as well. Some symptom onset for the systemic events were anywhere between 2 to 4 days, and the resolution was anywhere between 1 to 2 days for each event. Symptoms tracked included fever, fatigue, headache, nausea, muscle and joint pain, vomiting, and diarrhea. And you can see the categorization. Out of all these systemic events, the most significant was fatigue and headache. Headache, which is not an unheard of finding on a pregnant mom, especially fatigue is, as you can see, more commonly detected in recipients of placebo than we did in the RSV pre-F vaccinated mothers in our study. If I move to the next slide, this slide is showing hypertensive disorders and the premature delivery, which was balanced across the RSV pre-F and the placebo group in this maternal participants. We are looking at the relative risk here, especially for the gestational hypertension, preeclampsia, and premature delivery. Two of our maternal participants with gestational hypertension had persistent postpartum elevated blood pressure, and they were ultimately got diagnosed with essential hypertension. In this study, we defined the premature delivery as delivery defined before 36 weeks, 0-day gestation. But as you can see from this table, no other adverse events of special interest were reported in the maternal participants. This was the entirety of the hypertensive disorders, 12 participants in the RSV pre-F and 15 participants in the placebo arm as well. And the premature delivery breakdown was 15 participants in RSV pre-F and 13 participants in the placebo arm with a relative risk of 1.14 in between two arms. In the next slide, we are moving to the adverse events after birth for infants. These are reported through 1 month after birth. They were similar to the moms, comparable across RSV pre-F and the placebo arms. And infant groups and the breakdown of the number of participants across the RSV pre-F and placebo were very balanced. And if we look at the newly diagnosed chronic medical condition, which has been a topic of interest or an adverse event of interest for us, there were one on each arm, and one RSV pre-F baby got diagnosed with eczema, and the other placebo participants had a HIV positive test within the 1 month after birth, and those were 2 of the newly diagnosed chronic medical conditions that's been reported within the 1 month of vaccination in infants. If I move to the next slide, you are looking at the adverse events throughout the study in infants. As you can see, the breakdown is very similar. In addition to our eczema baby, we had additional HIV positive test from another infant during this longer follow-up period, but the rest of the adverse events were 67% in RSV pre-F and 79% in the placebo arms, and they were balanced across the arms with no signal being detected as an imbalance. Next is the exploratory immunogenicity in endpoints, and let me emphasize one more time, this was our exploratory part of our study. We defined these endpoints descriptively. They were not powered to show statistical significance, but you will notice that numbers are pretty clear and robust. And this slide summarizes that in delivery, HIV-infected exposed participants show a robust immune response described as similar to non-HIV exposed participants. Let me orient you to this particular slide. On the left-hand side, you are seeing the maternal participants at 3 groups, my apologies. The gray one is the Marisol, the current trial I'm presenting to you, HIV-infected, exposed placebo group. The teal, however you want to call this color, teal blue color is Marisol HIV-infected RSV pre-F group. And magenta, this pink color is our Matisse participants. Those are non-HIV exposed, non-HIV infected comparison group from our pivotal Matisse trial. And as you can see, at the time of delivery, similar to our HIV non-infected, non-exposed participants in Matisse, Marisol HIV-infected exposed RSV pre-F group showed a robust immune response similar to the pink column here. And if you move to the right side of this graph, these are the babies, infants at birth. The gray ones are placebo group with their GMFR, geometric mean fold rises here, and the titers on rises on the top, titers in the graph. And this is our RSV pre-F, vaccinated moms, infants, and this is our Matisse, historical Matisse moms from South Africa who are HIV negative. And again, in these tables you can find the GMTR as well as GMFR and their geometric mean titers as well, it's presented in the graph. In this next slide, we are looking at the neutralizing titer GMTs with 95% confidence interval by visit. And this is depicted by our evaluable immunogenicity population in the pregnant women with HIV. It is a robust response to RSV pre-F at delivery. You can see the before vaccination for RSV-A and RSV-B here. The blue versus gray placebo, and the last column is showing, or the last part of the graph is showing the RSV-A-B combined, which is calculated as the geometric means of the RSV-A and B neutralizing GMTs for placebo versus RSV-A-B, and we are seeing 7-fold rise here at delivery within our vaccinated mothers. And on the next one, now infant titers at birth, infants of the RSV pre-F recipients show robust neutralizing titers too. These are infant GMTs with 95% confidence interval at birth. evaluable immunogenicity population. GMTs at birth for RSV-A is 0.99, for placebo it is 1.01, but after vaccination we see that the changes, at birth this is significant for the babies when you compare the placebo versus vaccine arms for RSV-A and RSV-B as well as RSV-AB combined. Again, RSV-AB is the geometric mean of A and B GMTs. Metric is the 50% neutralizing GMTs for this particular graph. This is a descriptive comparison, maternal response with HIV versus without HIV, using Matisse as a comparator. This is an exploratory descriptive immunogenicity analysis from the South Africa, and for the GMR of RSV-A and RSV-B, as you can see, the values are 0.91, 0.82. This is slightly lower than the Matisse participants by the GMR value, but this is something we expect from HIV positive individuals when it comes to their immune responses. Probably making the statement to a group of immunologists and physicians is risky, but I will just go ahead and say that this was a slight decrease from the HIV negative population we were expecting, but still by the measurement and the values we can comfortably call this a robust response to RSV-PREF in our HIV positive mothers when we compare it to the Matisse participants. And the HIV-exposed infants display this lower RSV neutralizing titers when compared to the RSV unexposed infants. We connect this to the same phenomenon. It's essentially, in many cases, linked to the transplacental transfer, being impacted by HIV infection and chronic inflammation in mom. But same ANCOVA approach, we just adjusted 6, and the baseline titers here is a log scale, and these GMT ratios are still robust to see that we are seeing significant responses and transplacental transfer to these babies in RSV-PREF arm of the trial. This is little bonus information. This additional modeling we did in infant titers through 6 months, which is the half-life of 42 days, has been mathematically calculated by our stat team. And this figure models infant titers from birth through 6 months, and again, the assumption of the half-life of 42 days. Outcome modeled is RSV-A and B combined, 50% geometric mean neutralizing titers by month. And the curves compare Matisse non-HIV exposed RSV-PREF to Marisot HIV exposed and Marisot HIV exposed placebo across the 6-month time frame. And you can see the graphs are the slope is essentially following the same pattern, and this was a good visual representation of responses in our Matisse versus Marisot versus our placebo group. So if I move to the conclusion that RSV-PREF in our trial has been well tolerated with no safety concern identified in the study, the immunogenicity in the mothers living with HIV showed a robust immune response, while infants showed overall lower neutralizing titers compared to the non-HIV exposed infants, but they were still considered robust by the design. Implication essentially is that results could support the indication of obesity in South Africa, especially the including pregnant women living with HIV, which is historically a underserved and understudied population. Bottom line is that safety appears to be comparable to RSV-PREF and placebo. Maternal immune responses are robust, and infant titers, while robust at birth, slightly lower than the HIV negative ones, but we still conclude that this will provide a support to maternal RSV vaccination in South Africa with HIV positive and potentially immunocompromised populations in general. So, thank you. I'm going to pause here, and I will be happy to take any questions you may have. Anything that wasn't clear, I will be happy to answer.
Thank you, Dr. Rassan. Thank you, Dr. Rassan. Can you listen us?
Yes, I can hear you.
Thank you very much for your talk, and congratulations on the very positive results you've seen so far. So my question is whether you have any plans on analyzing mucosal antibodies, antibodies in the mucosa, or whether currently the sera titers is enough correlate of protection for RSV. Thank you.
Thank you for your question. Correlate of protection analysis for MATIS and as well as this trial has been proving to be very challenging. We did not do any further analysis in this particular trial for any mucosal analysis or additional testing. But in our post-marketing studies, we are looking into breast milk antibodies, which can be very significant and critical in the pregnant moms, as you may agree. But just by the nature of this trial and being a more vulnerable population, no breast milk samples has been collected in Marisol trial. And when it comes to correlate of protection, one of the practices, one of the evaluations we are doing is comparison of this neutralizing antibody titers and or IgG levels to monoclonal antibodies similar to palivizumab or nirsevimab. And in the RSV field, it is the established value of the protective level, especially with the palivizumab. Nirsevimab studies are still ongoing, but we are looking at, actually I'm just going to go back a couple slides. In this particular slide for palivizumab levels of 100 microgram per milliliter corresponds to somewhere near 1,000 neutralizing titer for RSV A and B compared. So whenever, which is usually about a month time frame for the placebo recipients, they fall below that level and they lose any potential protection if they are not vaccinated, any potential vaccination from their previously exposed mother around 1 month. But for our MATIS as well as Marisol trial, for participants to come down to that monoclonal threshold level, which is essentially an indirect correlate for us, takes approximately 6-month time frame, which is the window for the vaccine protection and the coverage currently.
Understood. Thank you very much. And what about exploring T-cell response? Is that too challenging as a possible analysis of correlative protection?
So we did not have the T-cell response with the PBMC collection and the T-cell responses. That was a couple ongoing studies and analysis in our MATIS trial. The unfortunate thing is that how much blood you can collect from a clinical trial participants is always limited, and at one point in time those samples become very precious. So I think we are at that window. But the current idea is that going back in time, and as much as our sample availability allows us to evaluate and compare with this one, but those studies are still, evaluations are still ongoing.
So I have a question here, and this slide is perfect because here you show that there is no difference in neutralization between HSV non-exposed and exposed. But my question is, have you looked at deeply characterize the serum from these two cohorts, like in terms of antibody affinity or differences in probably isotype production or downstream effector functions between these two groups? Have you looked at that?
Yeah, if I heard you correctly, you are asking about receptor affinity analysis. Those are not done for neither Matisse or Morisot trial. The reason for that was both of them are designed as an efficacy trial, and immunosubset was limited with that sub-analysis we can perform. That's something to pencil down and look at receptor affinity, but unfortunately I don't have a data I can share with you in that regard.
Thank you for your talk. That was very interesting. I have two questions. First, whether therapy for the mothers could modify the results, or the women were under therapy for HIV. And the second question is, I think one of the conclusions was that there was no difference between the neutralizing antibodies for the mothers, but for the babies it was reduced. What would be the mechanism for that? Having the babies with lower levels of antibodies, but not the mothers?
Yeah, I'm going to start with your second question. Hopefully I heard it correctly. Mechanism that may be the reason for slightly lower responses on the HIV positive infants. But did I hear your question correctly?
Yes, it was in your conclusion slides.
In the infant side?
Yes.
Yeah. So, go ahead.
Yeah, if you can show your slide, the conclusion slide, because it was stated there, I think.
Yeah, this is the lower neutralizing titers, and again, our I should say theory, or the concept here is that in general, when you look at the HIV positive population and their overall responses to vaccine, which is a very well-known phenomenon that they do show less efficient transplacental transfer when it comes to the vaccines from mom to the infants. I think there are a couple of historical, I will be happy to dig those out for you, historical studies with the Tdap vaccination to infants born to vaccinated mothers. And actually, those were not in my slides, but one of the promising things we've seen in our trial is that study demonstrated efficient transplacental transfer. That was the interesting part in this case. Infants born to vaccinated mothers had higher titers at birth compared to placebo, and the transfer ratios were 0.99, which is like almost identical to what we saw in Matisse for RSV-A, and 0.86 for RSV-B. So RSV-B is always the problem child in between two strains. So that was surprising. But the mechanistic explanation of this is very likely based on the underlying chronic condition and the ongoing inflammatory process. Even in these mothers who we did not have active, elevated viral count at the time of vaccination. And our criteria require them to be stable for, or in the HAERT for 90 days. And the African guidelines says 2 measurements 2 weeks apart with less than 1,000, then they are considered responsive to the treatment. But even with those parameters, we cannot completely eliminate the fact that these mothers have depending on their length of HIV infection, ongoing and constant inflammation in the body that is present in multiple, cycles and the lifetimes. That is the main theory based on our general understanding of this, how vaccines indicate HIV positive moms. But with that being said, still with these ratios, the protective levels, especially neutralizing titer levels, remain significantly high in these infants, similar to, especially based on our modeling. We did not get concerned about them falling below protective levels by the 6-month mark.
Thank you very much. So there are more questions? Right.
I was impressed that the viral load was positive in some patients at the delivery. What was the problem? Adherence or the type of treatment?
Unfortunately, we had a couple of participants out of this 343. So it's right here at the time of delivery. As you can see, we had approximately 48 participants here detectable. Those were usually, they were uncompliant with their treatment regimen of the HIV, the HART treatment. In South Africa, they do a very impressive job, very efficient job following these mothers, making or in general, HIV positive individuals, they have access to medications, especially in this clinical trial setting, significantly easy. But unfortunately, the compliance is a different issue, and we did have a couple compliant participants that they became detectable during the time of delivery, unfortunately.
I understand. Thank you. Any other question? Great. Thank you, Dr. Nathan.
Thank you so much. I really appreciate the time you provided me today and gave me the chance to present this data. Thank you.
Thank you. Bye.
Bye-bye.
No sé si el doctor Galergi quiere dar una palabra para el cierre del curso. Bueno, quiero agradecer primero a la asistencia por su fidelidad que hace hasta ahora y su participación y su alegría durante los cafés. Y quiero agradecer al Instituto Milenio, a nuestros invitados y por las estupendas charlas que hemos tenido y realmente ha sido un día maravilloso que realmente he disfrutado plenamente y creo que ustedes también. Así que muchas